Welcome to the second day of the Janelia 40 Cellular Physiology workshop on the mechanics of physiological systems. Thank you for joining us. I am Margaret Gardel from the physics department and School of Molecular Engineering at the University of Chicago. Yesterday, we had 4 amazing sessions posing opportunities and challenges of understanding mechanical systems that drive physiology at nano to micrometer scales. If you missed any of those, they will be available via the Janelia YouTube channel. For today's sessions, we encourage the same level of interaction from our audience and panelists. And a reminder for the audience to please ask questions in the Q&A field. Panelists will need to use the chat function. I'd like to now turn this over to my co-workshop organizer, Daniel Goldman.
Hello all, thank you again for coming yesterday and for joining us today. It was on behalf of myself, Margaret, Wyatt, Korff, and Gwen Card, and Janine Stevens supplying and keeping the meeting running beautifully. It was a lot of fun coming from the biomechanics side of the world to see common features and principles and topics, force, phase transitions, control ideas at the micro and nano scale yesterday. And I'm excited for the folks thinking at larger scales to demonstrate these ideas today as well. So with that, let me hand the program over to Kiisa Nishikawa and Jim Vigoreaux, who will take over and start our session 5, muscle intrinsic dynamics across scales. Take it away.
Thank you, Margaret and Dan. And as Margaret mentioned, and those of you who were with us yesterday were treated to fascinating talks on cellular and subcellular mechanics and the various ways in which subcellular forces can be seen, measured, modeled to elucidate their role in cellular physiology and architecture. The talks today will venture beyond the boundaries of the cell to further explore the role of forces across scales of biological organization and complexity. So the 2 major themes that you will hear today are scales and integration. Biologists like myself who are interested in mechanics yearn for an understanding of a fundamental quality of living systems, namely movement, which in animals scales up to the level of the organism in various evident forms of locomotion, such as crawling, running, flying, swimming, just to name a few. This understanding requires a convergence of bottom to top and top to bottom approaches, an integration across scales of time and dimension, and examination of the interaction and interplay of systems within the organism and between the organism and the environment. So with that said, I'm pleased to introduce our first speaker today, Malcolm Irving from College London.
Hi, thanks, Jim. Let's just see if I can start the timer. Okay, and share my slides. Thanks. Can you see that?
Yep, looks good.
Okay. So I'm going to talk about the control of contraction in the heart as an example of muscle intrinsic dynamics across scales. The function of the heart can be understood, of course, only in the context of it being a component of the circulatory system. So in other words, starting at the level of the organism, where we have good functional readouts of heart performance in terms of blood pressure, cardiac output, and heart rate. Turning to the heart itself, the anatomy is quite complicated, but again, we have very good functional measures in terms of ejection volumes and heart rate, and the link between the organism and organ level in the heart is very well defined, even in individual, because of the wonderful things you can do with MRI these days. Going to the cell level, the muscular walls of the ventricle in the heart contain locally aligned muscle cells. So at the cell level, if we zoom in, we can see this electrically and mechanically coupled syncytium, which means that the wall of the heart operates synchronously and in a mechanically coupled way. That yellow line there is the junction between two cells. So we can describe cell force and cell length in quite a well-defined way if we know what the whole heart is doing. Similarly, these structures you can see on the fluorescence micrograph are the sarcomeres in the cells, and that means the monomers in the heart muscle cell. And then if we have information about the number or fraction of motors attached and information about the structure of the motors themselves, we can calculate things about force per motor, motor stroke, and biochemical rate constants. And then knowing some information about molecular structures, we can go to the level of chemistry, free energy, and molecular dynamics. So those are the length scales, and the heart is quite prescribed across those length scales. Turning to time scale.
Malcolm, you're coming a little bit broken up across the ocean.
This is a very familiar.
If you might want to turn off your camera, that may help a little bit.
Yeah. Okay, I'll do that. But in order to do that, I need to get that menu. Okay, so this familiar diagram shows the function of the left ventricle in terms of how the pressure and the volume of the ventricle vary with time. The electrical activation triggers a contraction of the ventricle. So there's a very rapid increase in the pressure in the ventricle and the stress in the ventricle wall. This only takes about 50 milliseconds and occurs at constant volume. The pressure builds up until the aortic valve opens, and then the ejection phase starts. You can see the volume decreasing in the green curve as blood gets ejected into the aorta. Then as the pressure in the ventricle becomes less than aortic pressure, the valve closes again. The heart very quickly and quickly, as it must do, in order to be for efficient refilling for the next beat. I'll come back to this diagram in a minute, but first I want to put this time scale of the heartbeat in a wider context. That context is the time scales of different types of regulation in the heart. So the fast regulatory processes, which I've called intrinsic regulatory mechanisms, occur on the time scale of the heartbeat. This is the sort of built-in level of control at the sarcomere, including calcium signaling to the tract, to the actin filaments, motor-based regulation, which is something that's dated relatively recently, and intrinsic length and load sensing, which is only just starting to be clear as important mechanisms. On slower timescales, we can consider systemic regulatory mechanisms, by which I mean, the best example here is the increase in cardiac output and heart rate during exercise. This is now the heart responding to systemic signals carried either by nerves or circulating small molecules, and intracellularly translated by post-translational modifications of proteins or other proteins inside the muscle cell. And then finally, on the slower scales, is the class that I've called transcriptional regulated modeling, which is what happens in normal development and aging, but also in heart disease of various types and the many, many triggers that control those processes. Of course, all these three levels of control are not completely independent, but linked, and especially it's helpful to know something about the intrinsic mechanisms in order to understand more about the slower mechanisms. So I'm going to concentrate on the intrinsic mechanisms. And going back then to the scale issue, and starting with my original diagram of the heartbeat in terms of what the organ does, we can translate that into what happens at the level of the filaments because the anatomy of the heart is sufficiently well known. So within the sarcomere, within the myosin filament, the stress in the filament rises to about 100 piconewtons at the peak of contraction in the heart, with about the same time course as the wall stress in the ventricle. So this is the synchronicity of the heart, which means that you can easily relate molecular level events to organ level events. And similarly, you can see that the ejection phase of the heart corresponds to filament sliding of about 100 nanometers. So that means that we can take those parameters about intrinsic regulation and say, well, how do the myosin motors know how to behave like this? And they have to gives the start signal. But how do they know when to relax, and how do they respond to changes in external conditions of the heart like stress and length? And that's where these intrinsic mechanisms. And I've already mentioned the well-known calcium regulatory pathways that signals to the actin filaments. So in addition to that, that myosin motors can be inactivated independently of the calcium syn filament pathway. And indeed, at this transition, they're inactivated by the motor domains folding back against the tail, just like in smooth muscle. So this actually happens on a beat-to-beat basis in the heart. The motors are coming out to bind to actin and then being parked again at the end of the beat. The regulatory states of the myosin and actin filaments are positively coupled, and that therefore controls the transition that I showed in the previous slide. And we're starting to learn a little bit about the mechanisms for that. And here's one of the candidate mechanisms involving this protein called myosin binding protein C, whose N-terminus can bind either to the thin filament, the actin filament, or to the myosin filament, and so can shuttle in between and signal between filaments. Another intrinsic mechanism that we know a little bit about, filaments can be directly activated by stress. There is myosin filament mechanosensing. So if you pull on the end of a myosin filament, leave the folded off state, come out, and can interact with actin. And we're starting to understand a little bit more about that mechanism. But in the heart, what that mechanism would do would be that tunes up the motors when they have to work against a higher load, like for example, in response to higher aortic pressure. And then the bit that we know least about, which is length sensing, the very strong evidence that myosin filaments can also sense sarcomere length, although we don't know how. This was known 100 years ago by physiologists. They knew that increased venous filling in the heart leads to a stronger contraction in the next beat. It's called the Frank-Starling law of the heart, or at the cell level, it's called length-dependent activation. We're starting to learn about the mechanisms. And possibly the reversal of length-dependent activation is that when you have shortening during ejection, it automatically switches the heart off, which is obviously another desirable feature. So finally, I just want to briefly address the tools question. What tools and systems, model systems do we need to understand these things better? Well, of the rat. That's not an ideal model for them. We to have better models that mimic better. We'd also like to have better work across different time and length scales. Time resolved is a diffraction pattern from you can measure this with 10 million time resolution. And this is the fluorescence method that we use to follow the change of regulatory domains of proteins. So these methods have some limitations and we need to overcome those. And finally, we need computational models that can help us bridge the scales. I've oversimplified it in my introduction. It's a bit complicated than that. And we need models that will allow us to extend these studies of healthy heart to heart models of heart disease, which we can then use to understand the mechanisms of disease and develop therapies. So with that, I'd like to thank all the people who've been working with us on these problems. I've talked only about heart muscle today, but we also work on skeletal muscle. These are all the people who are involved. So thanks very much. With that, I'll open it up for questions. Thank you. And I'll try.
Thanks so much, Malcolm. That was just such a beautiful talk. There's one question here from Virginia Rutten in the question box. Could you comment on the role of intrinsic cardiac nervous system? What parameters is it thought to affect or control? So maybe a little bit more about the intrinsic control of the heart.
Yeah, you know, that's something that I know very little about, because we've taken rather a reductionist approach. I think that's it's an example of the extra things that we will need when we try and build back from this reductionist approach to understand more about how the heart works, especially on slightly longer timescales. I suspect that it's intrinsic to the heart, but it probably is not working as fast as the heartbeat, or I could be corrected on that.
Manu Prakash asks, what possible hypotheses for length sensing are currently being explored?
Well, the favorite hypothesis is controversial. The system works approximately volume, so the distance between the filaments also changes, so that when the sarcomere shortens, the filaments get further apart. So one hypothesis is that something which senses interfilament spacing. Hypothesis, but actually it's something we're looking at again, and I think the published evidence, which has been generally accepted as not supporting that hypothesis, needs another look. So at the moment, that's actually my favorite hypothesis, although it's very speculative at the moment.
Wyatt asks, can you comment on the value of studying muscle across species? Sorry, there's an airplane flying overhead. How common are principles of stretch activation in flight muscles in flies and cardiac muscle?
Yeah, good question. Yeah, there's this fascinating parallel between stretch activation in asynchronous flight muscle and in the heart. And we don't really expect stretch activation to be of much physiological significance in the heart, although again, if you think about how the ventricle contracts in 3 dimensions, there's a possibility that during certain phases of the contraction that the epicardium or the endocardium stretch. So that may well come in at that level. The more general question of the value of comparative studies, I think they're extremely valuable. There are very clear differences between the small rodent models that we use and large mammals and ultimately man. And it's very important that we understand those to understand the heart. But then similarly in skeletal muscle, the differences can be very informative.
So Jim, how are we doing on time? I can't unfortunately see the timer.
I think we're about 5 minutes into Q&A, so I think it's time to move on to the next topic.
Okay, well, unfortunately, there's quite a lot of interesting questions remaining in the chat and the Q&A. So Malcolm, maybe you can have a look at those and answer some of those offline. Thanks again for the lovely talk.
Thank you.
Okay. So apologies, I had to log out and relog. Momentarily lost my connection. So our next speaker is Flavio Fenton from Georgia Tech.
Hey, thank you. Thank you so much for inviting me. Can everybody see my screen? Can you hear me?
Yes.
Yes.
Okay, good. Thank you. All right, thank you again for inviting me to such a great workshop. So Malcolm did a good job presenting some of the basics of hard dynamics and temporal changes. I'm going to talk more a little bit about the scaling in space more than in time. So as we were saying, the cardiac cells are about 100 microns by 50 by 20 microns. They are like elongated cardiac cells that contract. They contract because you have the membrane, and every time there's a current passing through that membrane to ion channels that open and close as function of voltage and time, then change the membrane potential, and the membrane potential changes, allowing calcium to go inside the cell and make the contraction of the cell. So the electrical signal is basically driven by Ohm's law with all these ion channels that go through the cell. The important thing is the conductance is a very highly nonlinear conductance that depends on voltage and calcium and time and space. So there are different ways to model these. They can be from simpler models that we physicists use of a few variables to models for biomedical engineering with up to 100 variables for a single cell, and then you couple them in space. But the main idea is that you have to model then the electrical signal. So every time the cell contracts is because there's an electron signal that we can see here, and that's called the action potential. And it changes the voltage from the inside to the outside of the cell from minus 80 millivolts to 20 millivolts. So it depolarizes the cell, and then it repolarizes the cell. When the cell gets depolarized, then that triggers calcium to go inside the cell. Calcium goes inside the cell, gets the contraction with the myosin actin, make the contraction, and that makes the cell contract. So when you think about, and then in tissue, then the whole heart follows the whole contraction. So when you think about single cell, it's just basically little oscillators that they can oscillate, and then they can be out of sync or in sync, depending on the normal dynamics or an arrhythmia. But the nice thing about the heart is that it's a very nice example of going emerging behavior as you go to in space. So from single cell, which is considered 0D, we can go to 1D. The heart, when you open the heart, this is an open view of the heart. You can see the atria on the top, the ventricles on the bottom. You can see these little bridges here. This is called the Purkinje fibers. So those fibers send the electrical signal for the contraction of the heart, try to send it almost simultaneously to everywhere. So there are fast conducting cables. So there are actually one-dimensional cables that you can take out, and in experiments that you can actually measure an electrical signal propagating through the cable. So, and every time I talk about an electrical signal, there's a contraction signal following. So you have these electromechanical waves. So that's what happens in single cell. I mean, in 1D, you can have waves. In 2D, you can have, you can consider the atria. It's a very thin layer of atria. So the atria can be considered a quasi-two-dimensional tissue. But people can also create monolayers, put in many of the cell layers in 2D and create two-dimensional experiments. And when you go to 3D, then it's a very complicated structure when the fibers that generate are formed by the cells, they actually rotate 180 degrees as they go from epicardium to endocardium. So that there's a complex anisotropy of the wave propagation in tissue that then we have to study about. So just as we go from a single cell, when your cells, the only thing they can do is oscillate, once you couple them in space, you can create a one-dimensional tissue that you can actually create waves when you press. So this is one simulation of a one-dimensional tissue. If I stimulate at one side, the cells are going to propagate and you create an electrical wave. And following this electrical wave, there's a contraction wave. One interesting thing that these cells have is that you simulate in the back of the wave is refractory. If I stimulate back in the wave, then I can excite again. So there's a refractory period behind a wave that affects that you cannot excite below. The interesting thing is that if I stimulate then and create a wave that propagates, if I create another wave that creates on the different direction, when two waves collide, they actually annihilate. So this is different than when you have sound waves or water waves that they pass each other. Electrical waves and contraction waves, when they collide, they annihilate each other. All right. So when you go to 2D, in 2D now, these waves actually have another imaging behavior. So you can have an electrical signal. Let me just change this different setting. So I can stimulate all around this area here, and create and initialize. So I can create a wave, a one-dimensional wave that propagates in 2D. But what happens if I stimulate in the back of the wave? Then in this region, it's going to be refractory, but on this region where my mouse is going to be propagating. So I can break the symmetry, and in 2D, actually, I can create spiral waves. So those can form in the heart. That's an arrhythmic wave because it propagates much faster. And these waves, there are actually many different mechanisms here. I'm just doing it by hand, but there are many different mechanisms can break the spiral waves into multiples, and you can have multiple spiral waves behaving in the heart. That changes the rhythm of the heart and becomes arrhythmic. So once you have spiral waves, actually, the spiral waves depend on the parameters of the system. Can have a regular, like you can imagine all the time, a single spiral wave with a circular core. But as you change the parameters of the ion channels, you can go to a cycloidal trajectory. You can create a cycloidal trajectory, a hypocycloidal. It can become more what is called hypermeander. That is a very complicated trajectory of the spiral waves. And then when you have very high excitability, you get what is called linear core trajectory. So these trajectories go from low excitability on the tissue to high excitability on the tissue. And we can observe those ones actually in experiments, and we can see them happen in the heart. So optical mapping experiments allows you to see these spiral waves, sometimes with, there are kind of pin spiral waves, circular core, but they can also show the other regime where we have these long line linear core trajectories that happen in the tissue. The important thing about these spiral waves when they happen, this is an optical mapping experiment on a pig heart. And you can see that when you go from normal rhythm, when you initiate a spiral wave, that forms what is called tachycardia, and that makes the heart contract at a much faster rate. And then these spiral waves can break into multiples, like in this case, and then create fibrillation. And that's sudden cardiac death. And the only way to terminate this arrhythmia is by electric shock that brings back the heart again to normal rhythm. Now, this is everything I showed you, just 2D, but in reality, the system is 3D. The heart's a three-dimensional system. Once you go into 3D, a two-dimensional spiral wave becomes a three-dimensional scroll wave. So that means that at every plane, you can follow the tip of the spiral wave, and it becomes a vortex filament. And if you follow this vortex filament, just by calculating the curvature and twist, it gives you information about the direction of the spiral waves. Now in 3D, because you have a vortex filament, now you have a lot of topological effects as well, because this vortex filament can actually, the tip of them, they can touch each other, so you can form a scroll wave, a ring. You can create a ring of rings, so the vortex filament, the spiral waves can go from surface to surface, or it can be intramurally. And also once you are intramurally, you can have new scroll waves appearing and disappearing, like in this case, in the top box. They can appear and then join to each other if they have the right phase, or one single spiral vortex filament can elongate and pinch itself and produce more another vortex filament. So that's new topology happens in tissue. That's more complicated. And then when it happens in tissue, you actually can visualize the complexity of these vortex filaments creating when fibrillation appears in the heart and produces multiple waves and arrhythmias. So I didn't follow I couldn't see my timing, so I don't know what my timing is. Hopefully I went in time. My main conclusions here is just to show you this short talk, the complexity of the dynamics of these electromechanical waves that happen in the heart. They grow with dimension due to this emergent behavior. As you couple tissue, it's very interesting how you see the new as you increase the dimensionality, and so you can still have the dynamics that you have in the previous dimension, but as you increase dimension, you can expect to have new dynamics that you didn't observe before. I would like to thank all these, a lot of this work has been done for a lot of my students over the years, and they're really, really good. And I have some support from NSF and NIH. Thank you so much. I'm open for questions now.
Thank you for that fascinating talk. Flavio, we have some questions for you in the chat. The first question, just disappeared from my screen, was from Jimmy. Where did it go? What is the mechanism underlying the destruction versus superposition of intersecting waves?
It's just basically the refractory period. So after a cell gets excited, all the ion channels have to recover back to normal before they excite again. So this is a typical example of a fire. So that's why waves, fire, you fight fire with fire, because you have an electric, you have a fire wave that is burning tissue, or burning grass, but behind the wave, there's no more grass to burn. So if you want to excite another wave, you have to grow the grass again before you can create another wave.
Thank you. Second question is, to what extent does coupling between electrical excitability and mechanics dynamics of contraction shape spiral wave dynamics?
Yes, that's a very good question. So it can change a lot. The excitability of the system will change the dynamics of the waves, and the contraction, as you start decoupling the contraction, it will slow down some of the electrical. So it's a very nice couple between space and voltage. So if you start decoupling the contraction, you start decoupling also the electrical, how quickly the activation goes, so you can slow down the waves and you can change the wavelength.
We have a question from Manu. And first of all, congratulations on a beautiful talk. Manu is curious if these simulations include heterogeneity in wave propagation because of anisotropy in the tissue.
Yes, so anisotropy becomes very important in 2D. So of course, 1D doesn't matter. In 2D, you can always rescale the system. Once you go to 3D, 3D then you have this rotational anisotropy. And that's a mechanism that we know and we have observed. We actually discovered first numerically, and then we observed it experimentally. This anisotropy of the fibers that rotate 180 degrees from epicardium to endocardium can destabilize spiral waves. And depending on certain parameters on the dynamics of the heart, these spiral waves can destabilize very quickly due to the anisotropy of the tissue.
Another question is, beyond the refractory periods, what other mechanisms are there to prevent or stop fibrillations?
Yes, so that's a very, very complicated question. There of course, so there are drugs, there are ways to terminate arrhythmias by drugs. So the drugs will change the dynamics of these waves. So you can try to either change the wavelength or change the excitability. So depending on not all arrhythmias are the same. So there are many different types of spiral waves that you can have and different types of arrhythmias. So depending on the type of arrhythmia, there are drugs that you can use. There's of course the electrical shock that you do. So the electrical shock basically excites everybody and terminates them. But there are other methods we develop low energy defibrillations. So when you use low energy to try to terminate the waves using nonlinear dynamics. So the nice thing about all this thing I show you is that the dynamics of the heart is chaotic. So you can have periodic orbits, and that allows you to choose when to stimulate or when to excite in order to do better at when to terminate spiral waves or not.
Okay. That question actually had another part to it. And the questioner is asking, it's just surprising to see how uncommon it is, given how easily they are to emerge. So do they often start and get quenched or never arise in the first place?
They never, so it's a very bistable system. So it's very nice. It's very easy. You can go between the one or the two, but it's very hard to happen. So you need to have a particular condition in order to happen. But once it happens, once you have fibrillation, it's very hard for it to terminate. It can terminate in smaller hearts. So there's a critical size of spiral waves to form. depending on the wavelength. So small hearts, they can be more, the spiral waves can form and then terminate by themselves, so like in guinea pigs and rabbits. But when you start going to cats and larger hearts, then once you initiate a spiral wave, it's very hard to terminate them. Unless there are some heterogeneities where the spiral waves can pin and things like that.
Okay, well, thank you for those answers. That's all the time we have for questions. I do encourage you, Flavio, to go to the Q&A box and answer any incoming questions.
Sure.
So our next speaker will be Bavat Bornstein from the Weizmann Institute of Science.
Okay, you see my, hello?
Yes.
Yes, good. So first I want to thank you for inviting me and present my work here in this workshop. It was very interesting and I'm looking forward for the discussion today. My name is Bavat and I'm a postdoc at the Zelzer lab. And in the lab, we studied the development of the musculoskeletal system. And what's mainly interests me is how the nervous system, and particularly how the proprioception system, interact and affect the development of the musculoskeleton. So proprioception, what it is, it's in a sense, it's our one way to say it is our sixth sense. It's the sense that's govern and sense our self-movement and the position of our body in space. And one test we can do for proprioception is really simple. We can close our eyes and try to touch our nose. Most people can do it with no problem. However, people with proprioception difficulties have problem doing so. In mice, it was shown that RANX3 transcription factor is expressed in sensory proprioception neurons, and mutation in RANX3 results in the ablation of proprioception neurons. As a result, you can see mice that have ataxic movement, which is uncoordinated movement. In the lab, we showed that these mice also exhibit scoliosis, which is a curvature S-like shape in the spine. And this raises an hypothesis that maybe proprioception can also regulate spinal alignment. Actually, we in the lab have shown that proprioception not only affects spinal alignment, but also affects fractured bone repair and normal joint development. This raises the possibility that proprioception dysfunction may contribute to the etiology of various musculoskeletal pathologies. Therefore, understanding the molecular component of proprioception and understanding how can provide the understanding and insight into the mechanism underlying some of these pathologies. However, despite the importance of proprioception, the molecular identity and the mechanism that mediate its function is mainly unknown. And one way to study proprioception is simply by studying the proprioception sensory organs, the Golgi tendon and the muscle spindles. Between them, muscle spindle was shown to be the main proprioception sensory organ. This is why we're going to focus on, in this talk, on the muscle spindle. And a muscle spindle is embedded within the muscle. It senses the stretch and the force that is acting on the muscle. It's comprised of sensory neurons that innervates and wrap around specialized muscle fiber that is called intrafusal muscle, and everything is wrapped around a capsule made out of extracellular matrix and connective tissue. So we reasoned that if we'll study muscle spindle development and combine it to skeleton development, we might understand some of this regulation and might be able to answer some of the mechanism that regulates between those two systems. However, studying muscle spindle is quite challenging. It is embedded within the muscle. It's relatively small and it's not abundant at all. And it's a complex organ, like I said before, it comprises of different tissue, and the regulation between the different tissue is really unexplored. All of this results in the way that we don't have good markers to study the different tissue and understand how it functions. So in a way, you can think about it as like, where is Waldo? And in order to find Waldo, you need to know that it wears a red shirt with white stripes. So the first goal in our project was to find and understand what's the different tissue in muscle spindle wear in order to find them. It is Important to say that recently some molecular understanding of how muscle spindle develops and function has been emerged, and however, most progress has been done on the proprioception sensory neuron, leaving behind the intrafusal and the capsule and the rest of the tissue. So in order to gain understanding on the entire organ, we wanted to identify and study the intact organ. So for that end, I dissected out muscle spindle, intact muscle spindle, comprising of the neuron and the intrafusal muscle and the capsule, and sent them to transcriptome and proteomic analysis. And all of this analysis was done and compared to adjacent normal muscle. By combining both RNA and proteomic data, we were able to uncover 4 molecular markers for the different tissues that comprise the muscle spindle. We found intrafusal marker, myosin light chain 2. If those heart experts from the previous sessions can maybe identify it, know that is one of the myosins expressed in the heart. We found a marker for the neuronal part, marker for the outer capsule, and then extracellular matrix marker. And now, in order to gain a better understanding on the complex and the entire expression of the morphology of each marker, we wanted and we calibrated a clearing and staining protocol of imaging whole mount muscle using confocal microscope. And here you can see volume rendering of an example of each marker. And for example, you can now see the capsule GLUT1 staining, and now you can also understand how it expression is related to the other areas of the tissue. Now we have these markers, and we can start asking questions about how muscle spindle develops and how the morphology changes during pathologies. So due to the lack of time, I'm going to share with you the data that we have using the capsule markers. And we start with studying the development of muscle spindle and looking at the postnatal development when muscle spindle is during its maturation, looking at the staining of the different marker of the capsule, though we know that at P3, which is 3 days after the mice are born, we have capsule around the muscle spindle. We do not see any staining of the 2 different markers. However, at P7, we see that specific capsule, outer capsule is marked by GLUT1, and only at P25, we see both the outer capsule and the extracellular matrix markers are expressed. This indicating on a sequential step of muscle spindle maturation. Next, we wanted to use this toolbox of markers to study the muscle spindle in mutant mice, for a proof of concept and also for validation of our markers. We turned into the EGR mutant mice, which was shown to have a proprioception defect. And together, and also we showed that it has a skeletal alignment problems, and it was shown to have muscle spindle, however, they are degenerated. So using the capsule staining, we can see that in normal wild type mice, the diameter of the capsule is about 50 micron. However, in EGR mutant, we see that it's much reduced. And this correlates to the extracellular matrix staining that is also dramatically reduced, indicating that maybe the diameter of the muscle spindle and the morphology changes are due to the extracellular staining, the extracellular reduction in extracellular proteins. So we have those, we've shown that those markers are good to study muscle spindle during development and pathologies. And what next? We want now to go back to our RNA-seq data and find candidate genes in order to study and better understand the relationship between the proprioception and the muscle skeleton regulation. And we want to use the tools that we identify in order to visual muscle spindle, combine it with in vivo CT scans, and do functional proprioception test. And maybe we can get some more insight into this fascinating regulation between the different systems. Some challenges, and this is for the Janelia guys, what you can help us and to do to get better. So I would like to get a better cellular resolution on 3D imaging. And eventually I would also, it would be really good to see and understand how muscle spindle function by actually visualizing stretching. So that means that we want to go into live imaging. It means that we need to transform all of our staining and antibody into reporter lines. And when we go into live imaging, we also have problem, I'm talking about now, penetrating within the muscle and overcoming movement problem while imaging. So those are some challenges if you can help us solve. And going into the big candidate screen challenge is that we would ideally do this mutant screen on a conditional knockout in the precise tissue that it is expressed. So creating more tools for really a precise conditional knockout will be amazing. And if you can, guys, make the time of generation much faster, so we can be able to screen more mutants, that would be amazing. So with that, I will end, and I would say thank the Zelzer lab, especially Leah and Sharon, who did the dissociation and isolation of the spindles, and Sarah and Sveta, who helped me with the clearing protocol, Ellie, my mentor, the bioinformatic unit, and you for listening. Thank you.
Thank you, Bavat.
So we have
that was
we have extra time.
Very nice talk. Oh, here's a question in the Q&A. Is there any evidence for muscle spindle sensing the spine shape itself?
That's a good question. We still don't really know how muscle spindle or proprioception sense and affect muscle spindle, the spine. The spine itself is intact. So the different vertebrae are correct and in the right shape. It's only their alignment that is affected. So I think it's more regulation between the different size or regulation of the tension that this muscle sense that change it, but it's still not known.
Do humans with proprioceptive defects have similar skeletal alignment defects as well?
Yes, definitely. I was actually, one of the examples is a piezo2 ion channel. So humans with piezo2 mutants also have proprioception defects and have scoliosis and hip dysplasia, which is another symptom that we see also in our proprioception mutants. And I didn't have time to say, but the thing is that the mice, when they develop scoliosis, so they develop normally and only around their puberty they start to have. So it's really resemble the etiology of the idiopathic scoliosis that happens in humans.
So I have, I guess, a question. Oh, there's one. Yeah. So it seems like there's a lot of myopathies that don't involve proprioception that also lead to scoliosis. So we have a mutant mouse that has a Titan mutation and it has, a really kyphotic spine. So I resonated with what you said earlier about there possibly being indirect mechanisms that the proprioception changes the loading pattern of the muscles, which then changes the loading of the skeleton and leads to that sort of.
Yeah, again, the feedback mechanism between the different systems. So if you don't have a way to sense how much your muscle is being tension on, so you cannot correct it and accumulate to it in larger scales. So definitely.
So do these mice, one last question from me, do these mice have muscle pathology too? As in, for example, do they have, muscle wasting or other muscle pathologies associated with the proprioception?
I have to say that I'm not sure. I don't want to say something, and we need to check. Probably the RANX3 and the EGR mutant were characterized and checked for muscle pathologies. I know there are the RANX are smaller, but I don't know. But
Okay.
that's a good question.
How's our time? Is it time to move on to the next talk? Yeah.
It's time to move on to our discussion, yes.
Yes. Oh, right, we're on the discussion. Thanks again, Bavat. Yeah. Lovely talk.
So, yeah, we can just move on to our discussion. Thank you for the three speakers for very interesting and stimulating conversations. So, one of the themes that I saw across the three talks is this sort of a coupling or interaction or intersection between systems, namely the nervous system and muscle. So I would like to, and I know this has already been addressed in some of the questions, but maybe further explore that intersectionality of these systems and how they could influence one another. For example, to what extent does changes in electric excitability can be compensated or can influence the intrinsic mechanical behavior of a sarcomere, for example. So it's sort of this concept of a crosstalk or interplay between systems. So I'm wondering if any of our speakers or members of the panel will like to comment on those.
I could say that yeah, the nervous system has, of course, a big effect on the heart, but fortunately, the heart doesn't get tired, right? Like any other muscle, so it doesn't stop. But the nervous system, when it gets the empathetic and sympathetic nervous systems, when they act, what they do mostly is release certain compounds in the heart that then change the dynamics of the heart. So when you get scared, you get epinephrine is the liver, and your heart starts contracting much stronger. When you relax, acetylcholine gets the liver, and that changes a little bit also the wavelengths of the arrhythmia. So sometimes, actually, experimentally, if you stimulate the sympathetic nerve, you can initiate arrhythmias in the atria very easy. So those are some of the effects that you can have with the nervous system.
Yeah, Jim, it's interestingly different in the heart and in skeletal muscle. Flavio has described what the heart innervation does. Skeletal muscle is clearly totally different. And one thing that's very interesting to me, and I wonder whether anyone else would like to comment on it, is that it seems to me that the innervation in skeletal muscle is a kind of start or go signal. And has evolved really to be fast, which is to say, to produce the contraction as soon as you get the action potential at the motor end plate. And then all of those processes are very fast. It's interesting that the vertebrate muscles don't have any inhibitory input, as far as I understand it, whereas some invertebrate muscles have inhibitory innervation. So in vertebrates, the central nervous system has no way to say to a muscle, that's enough, stop, relax as soon as possible. And so I'm interested in what people think about whether that's because the CNS invertebrates evolved completely different types of control system, which probably rely on muscles working as agonists against antagonists. So the best way to stop an agonist doing something is to activate the antagonist. But I wonder if anyone would like to, I don't know much about the control theory behind all this.
Well, first, one way to control is, again, the muscle spindle. It sends the strength, and there is like instead of not only sensing and putting information into the CNS, it also have synapses straight on muscle motor neurons. And I think a lot of the regulation in vertebrates happens in not on the muscle itself, but in the spinal cord. So over there, you have a lot of inhibitory neurons that can regulate and moderate between the activation and inhibition of the neurons. So you have a lot of really complex inhibition and regulation in the spinal cord.
But what you don't have is any inhibitory motor neurons, which send stop signals to muscle.
One of the things that you were talking about, Malcolm, that I think is really important in skeletal muscle is the intrinsic properties. And I think forced depression with shortening, so the time course of events, we measure everything in isometric muscle, but the time course of events in skeletal muscle cells really depends on their ongoing strain. And so one of the reasons why skeletal muscles can relax really fast in behaving organisms is because they're shortening while they're deactivating. And this greatly increases the rate of shortening or of deactivation. And likewise, the rate of force development can also be modulated, over probably a factor of, 10 or 20 fold by whether you're stretching a muscle when you're activating it. So ongoing length changes in skeletal muscle really have profound effects on the time course of activation and deactivation.
Yeah, I agree with that. I think that at the end of a tetanus, if you've got a preparation with very low compliance in series with it, a skeletal muscle, it seems like it just gets locked on, and there's this so-called isometric relaxation phase, which was originally only described in amphibian muscle, but I think it's there in mammalian muscles as well. It's a catch-like state actually, where even though the calcium is low, the muscle just can't relax. And so, question is, how do you get out of that if you're really in an isometric situation? You need somebody else to pull you out, I think.
So I had a question for the discussion, and actually it was something that came up yesterday about universal models and understanding, systems at different scales. And I think universal models are impractical, but I think it's really interesting to examine the scale issue. And so my question for you and for Flavio is, what concepts do we need to add, when we go from trying to explain, heart dynamics from the level of the single motors to the level of the whole, 2D strips or 3D organs? So, how do the models at the different levels, support each other? How are they fundamentally different? Where are the conceptual gaps in between?
Yeah. Malcolm, you want to go first?
Yeah, I'll have a go. It seems to me that in the heart, Flavio has told you a lot about how the wave spreads across the heart, but I was kind of taking the sort of opposite extreme of emphasizing the synchronicity of different parts of the heart. And the heart, when it's at its most efficient, I think the more synchronously a particular chamber contracts, then the higher the efficiency is likely to be. And of course, a disordered, as you see with the extreme in fibrillation, disordered contraction is inefficient. My feeling is the heart evolution of the heart has been driven really to synchronize across scales as well. And I now see it that there's a direct read across between the very stereotypic set of events that happens in one heartbeat and what's happening at the individual myosin level. So that I started to think about the heart as a kind of biochemical single turnover myosin machine, that the myosins, you don't use all your myosins. You probably, ATPase measurements suggest that in a single heartbeat, you probably only use about a third of the myosins that are there in each filament. So each myosin probably only does its thing once, and the thing is synchronized with the heartbeat. So that's my view of it.
Yeah, so I agree with Malcolm that it depends on the question that you're trying to answer, right? So of course, there are some arrhythmias that are driven by voltage and some arrhythmias that are driven by calcium. So you may want to, depending on which type of arrhythmia you're trying to investigate, the type of modeling that you're going to use. And for some dynamics, sometimes very simple models, 3-4 variable models are enough to give you some information about the even bifurcations. So one interesting thing that happens in the heart is that it happens for mammalian hearts, but also amphibian hearts, and most hearts, when you pace really fast, they show a pure doubling bifurcation. And even though when you're pacing at a constant period, the response is not going to be constant. The reaction potential that you get can be long, short. So you got a period 2, then you can get a period 4, a period 8. That's one of the nice things about it's being a chaotic system. So those bifurcations, you can show them sometimes with very simpler models. How did they develop? What is the mechanism behind that? But all in the heart, there are some mechanisms that develop this bifurcation by pure voltage or pure calcium. So in order to make to understand what mechanism is relevant for a particular species, sometimes you have to go to a more complex model that describes the mechanism of the calcium dynamics in the system, right, or the contraction, because they're not all the same. So the interesting thing is that the evolution in the heart, the main idea, all hearts contract, but how they contract is actually very different between species. It's very interesting how different the cells are not the same. The way the cells get nutrients by blood is not the same. So for example, an alligator, you cannot fibrillate an alligator. Alligators are, no matter what you do, they always keep contracting and they don't fibrillate. But in other animals like a horse, you just basically look at a horse heart and it would just go into fibrillation. It's very easy. So it depends.
Yeah, I have a follow-up question to that, but I want to go to Ed Monroe, who has patiently held his hand out for some time.
Yeah, these are great provocative talks. I'm wondering, to what extent does architectural plasticity come into play on longer timescales in these contractile systems? Are there mechanisms, homeostatic feedback mechanisms that couple architecture to, that couple activity to plastic changes in architecture that operate on long time scales?
Yes, so there exists exactly what is called a remodeling of the heart. So when the heart starts getting into atrophy, it's very interesting how both change. The electrical currents in the membrane change, but also the structure of the heart changes. So there's a remodeling of both electrophysiological and structural remodeling. And then sometimes you have to investigate which is more important. Sometimes it's more the structural remodeling, sometimes maybe more important the electrical remodeling. And sometimes you can derive one. So when people have heart attacks, depending on the type of heart attacks, they can do certain pacing. They can put electrodes in a different part of the heart to pace, to try to resynchronize the pacing. And then with the resynchronization, then the heart starts to recover a little bit the structural as well. So it can go bad or it can be good depending on how you stimulate.
Yeah, I would add to that there's some interesting examples with cardiomyopathies that you can have hypertrophic or hypertrophic cardiomyopathy, and it seems like you can have a number of different mutations in many different proteins that can lead to the same remodeled phenotype. And one of the theories about that is that this is really to do with metabolic imbalance, and some of those proteins that get changed might be enzymes which have nothing to do with contraction or the regulation of contraction, but with muscle metabolism. So one of the ideas is that a sort of long-term imbalance in metabolism can signal these atrophy or remodeling cues.
In the skeletal muscle system, of course, you have hypertrophy and sarcopenia, and these pathways are very well characterized at the molecular level. But there's a lot of really interesting variations. And so one interesting one is that Henk Granzier made a mouse, a transgenic mouse with a very, very stiff titin. It took him 10 generations to make the one of the parts of the titin molecule, it reduced 75% of its contact length. These mouse muscles actually get more compliant when you activate them. mechanism completely unknown. They also have hypertrophy, longitudinal hypertrophy, but they did a complete, whole transcriptome sequencing and didn't find a single known hypertrophy pathway that was activated. So I think that these model systems can really, tell us how much we still have to learn about some of these systems, and the knowledge always seems more complete than we blissfully sometimes hope.
Okay, so we have Manu and then Tom.
So I just wanted to make a general comment based on what Jim, you said about, this is muscle and the nervous is working as a system together. And Malcolm, you brought up the notion of invertebrates. I'm curious how people think about what are the total number of things that we need to map. Maybe, I think I'm quite inspired by Gaspar Jaeckel's recent work on mapping all desmosomes in an entire organism, mapping every single muscle, mapping every number of the neuromuscular junctions. And what they find is quite intriguing is the fact that the desmosome positions become really important because the mechanical forces are also propagating. And I'm curious, I mostly focus on invertebrates, whether this notion of, stretch activation of these other muscles, they are in a totally different state because the mechanical information is also propagating along. So again, highlighting the role of smaller systems. And I'm just curious, in an ideal world situation, what would be the other parameters that we should be mapping? Especially, I think Tom had brought up extracellular matrix, but I'm just thinking about couplings because these systems are mechanically coupled. So any comments on that? And especially from a Janelia perspective, thinking about small systems really have a whole organism view, which is quite beautiful.
I think that, again, it depends on your, what your question is and how, you say small systems have really simple, you can ask question, but if you're talking about, at least from our point, muscle spindle, only we find it in mammalians. There is some, all animals have proprioception, but sensing exactly how this muscle is stretched and regulating on itself, and the really complex regulation between them that you lose when you go to small and simpler organisms. So it's really all depends on your question and what you want to find. And if you're looking for complexity, then you're going to lose some of it when you're going.
And I think it's really challenging because the moving parts inside sarcomeres are so small. And many of them are existing. I think I heard a comment yesterday about, the limitations of like cryo-EM. Yes, we can image very small things, but only when the structure is nearly identical so that we can average to bootstrap the resolution. And so, so the variation in structure is missed. A lot of things we'd like to map are existing right below that optical imaging resolution limit, unfortunately. Should I call on Tom?
Yeah.
Gwyneth has her hand up too.
Tom was first.
Shall I go ahead?
Go.
That was very good. I just have a question for possibly both Flavio and Malcolm, which is, I read recently that you can introduce a deficit in a cardiomyocyte using AAVs or other CRISPR technologies. And over time, the calcium signaling and the calcium fluxes change, even though you may be introducing a modification in the thick or thin filament. So there has to be some chitter chat between calcium signaling and force generation. And that is going on as a plastic adaptation. Are you aware of this? Is this something that's been resolved?
Well, I think there's many elements to the calcium homeostasis in the muscle cell, and some of the contractile proteins are very substantial calcium buffers. So it is possible, of course, that some changes that you go in and make that change other proteins actually have unanticipated effects on calcium homeostasis, because it's another one of those things that's in a fine balance between a whole series of pumps and channels and exchanges and intracellular storage. But I don't have a more specific answer.
Just a quick follow-up on that, which is my understanding is that whatever intracellular calcium buffering is happening, the calcium levels seem to change in a way that compensates for deficits.
That's interesting.
Okay, I think we've gone over time. Gwyneth, I don't know if you want to.
I'll just say one really quick thing. Organizer's prerogative. A really, really fascinating discussion. Thank you, session one. Great talks. I guess I just wanted to encourage everyone in the spirit of today, really moving towards a little bit more even that Janelia focus of, what role Janelia could play. As you think about answers to some of these questions, one of the things we're really interested in is what are the big, interesting questions that are still open in this field, really piggybacking on Kiisa's question about what are the gaps in our knowledge? So, when we think about modeling, I think there's been a lot of talk about, of course, the model or the system depends on your question. That's great. I think we can all agree on that. But I would urge you to even go past that to say, but this is what I think is the most interesting question. And therefore, these might be the types of model or the types of system, that Janelia should especially think of promoting or exploring. I think that this conversation started to maybe touch on that a little bit, and I'm really interested to continue the conversation as we go forward today.
All right, great. Thanks, Gwyneth. Let's take a quick break here. We will resume in 6 minutes at 12:20 Eastern. All right, I think we'll get started with session 6, integrative mechanics. And for that, I'm going to turn it over to the session moderators, Jimmy Liao from University of Florida and Wyatt Korff from Janelia. Jimmy Wyatt.
Hi, Jimmy, are you on? May have had some technical difficulties. So let me just go ahead and jump in and welcome everybody. Thank you. So this session is focused on integrating mechanics. They'll have a lot of overlap with the session 7 as well. And our first speaker is Bob Full from he's HHMI as well as the University of California, Berkeley. Bob.
How's the screen?
You're good,
Bob. Okay. Thanks a lot for this invitation. It's been incredible. And I want to talk about embodied control of behavior in complex environments. I'll give a brief trip along the control architecture.
Hey, Bob, I'm sorry to interrupt. There's a gray bar across the top of your slides. Is that something you could close out or move out of the way?
It's not on mine, but I think it might be due to the sharing issue, because I put on the ability to do videos. So let me share.
Maybe try one more time.
You have to take that off sometimes if you have this bar. And so,
should we introduce the session or just get rolling?
Yeah, unfortunately, I've lost the.
Should we introduce the session?
Okay, let me do it again. unfortunately, I don't see the.
That's okay. Why don't you
just go ahead and.
Why don't you reshare? Yeah.
Yeah, I unfortunately, as it is, I don't see the program to share, which is pretty odd. Yeah, it's very interesting. Well, I can try to do it this way. What does that look like?
That looks good.
That's fine?
Yes, it looks good. Thank you.
Okay, sorry for the delay.
That's all right.
All right.
Go for it.
So I'm going to talk about embodied control of behavior in complex environments. And what I'll do is I'll give a brief trip along the control architecture in animals, highlighting challenges with an example in each. Along with other legged runners, we run cockroaches on treadmills and quantify their biomechanics. And let me show you one surprising result. By using force platforms, we discovered that 2, 4, 6, and 8-legged animals all bounce like pogo sticks or string mass systems, where your one leg works like 2 legs of a trotting dog, or 3 legs of an insect as one, or 4 legs of a crab, bouncing like this kangaroo. Now, to focus our experiments, we follow the Feynman approach. We create both mathematical and physical models that serve as hypotheses. And yes, we know that all models are wrong, but a lot of them are very useful. Our approach really extends to components at lower levels of organization too, like muscle. And I show you this example because our mathematical modeling approach proposes a template, the simplest model of a system behavior that can serve as a target of control. The template's anchored in a more representative model to reveal mechanisms that you care about, maybe a leg or joint or muscles. And then we can collapse these dimensions back by trimming away the complexity. And we can do this at each level of organization because, as Bartholomew reminded us, every level finds its mechanism in levels below and significance in levels above. And so here's an example. In the upper left is the simplest spring mass template of an insect running down to the most representative anchor with 72 degrees of freedom. And each dynamical system collapses to the simpler system above. So that's a modeling approach. Now going back to the spring mass model, here's a plot of virtual leg spring stiffness that's normalized for weight and length. And we see that all-legged runners, from a cockroach to you, have the same relative leg stiffness despite this incredible diversity. And so we asked, could we use a physical model, a robot, to test this? And we used an interdisciplinary robot physics approach with the synergies with biology and physics and robotics. And after a significant amount of experimentation, varying leg stiffness, not something easily done in the animal, we better understood energy management and stability. And here is the robot Rex from Dan Kotochek's lab using this bouncing gait at 2 meters per second, and it's outside. Now, we here see only a small fraction of the literal zoo of physical models that people have built. And I think it's just the beginning of creating these. In this animal control diagram, the interaction with the environment is here down in the bottom right. And we ask one grand challenge is, how can structures interacting with external environments reveal function? So the organism and the environment really are inseparable. And we have to look at, of course, the natural environment, but I'd argue we also need to go beyond to understand capacity, because it may reveal a novel function that we just haven't seen yet. When great another physicist, Dan Goldman, came as a postdoc many years ago, he told me the ground mattered, and I wasn't convinced. And over the years, Dan and Chen Li, who will talk soon, showed that you can get no movement when this physical model is tuned to solid ground, but with a slight change, it can move pretty fast. And that's pretty convincing. With the help of Fei Kwan and Wyatt, thank you, Wyatt, we discovered a principle of foot design for really diverse creatures that we've been wondering about for a long time. Next, let's add the mechanical controller, the legs and feet and their interaction with the environment, and ask an important question of how do animals embody control using morphological computation? Here's one example. The dynamics of insects running in the horizontal plane can be modeled by a lateral leg spring template as they bounce also side to side. And our collaborator, applied mathematician Phil Holmes, discovered new mathematics as he gave us a novel model showing passive mechanical self-stabilization when perturbed while running. So sort of mechanical intelligence. Following Simon's work, thank you, Simon, we tested the mechanical feedback hypothesis by giving or running a cockroach a millisecond lateral impulse with a slight explosion and showed a very rapid recovery, faster than reflexes. We went on to explore the many parameters of passive dynamic self-stabilization in this physical model that you really couldn't do in the animal. So next, let's add neurofeedback with a simple reflex and ask the fundamental question, how does it work with the animal's natural dynamics? So here's an example. To better understand control modules, we measured a leaping lizard of a platform, first with pretty good traction, and then we made one with low friction. And so here's a control jump with the tail well-aligned body, jumping to its home. And this one's on low friction surface. The slip would normally cause nose down pitching, but lifting the tail counters the downward pitching through momentum trading with this very simple reflex. Angular momentum is redirected from the body into the tail with this reflex. And we tested this on a physical model we called TailBot. And here we were able to create a quantitative metric for a comparison. That led us to a more general conclusion about how you define the composition of control modules. Here we used a hierarchy of templates and anchors. So here on the top, you see just the body, and then it's anchored with just an appendage in the middle. But then anchors can reveal sort of the sheer design space for adding things like tails or limbs or even a flywheel. Since then, what's really important is research by Avik Day using a tail monopod hopper inspired by jerboas showed control module templates can actually be composed in both series and parallel to understand a suite of behaviors. And next, we want to do it on this animal. So last is the template selection on the far left. It includes deliberation and motivation, trying to decode an animal's representation of its own body and its environment. I think this is a grand challenge to explore the origins of creativity and innovation at the sensory motor limits of behavior. So why did we pick squirrels? Because they can do this. They're maybe nature's most agile animal making life and death decisions. There's a hawk who would like some lunch. And so we measure biomechanics of free-ranging squirrels on our campus with a platform. We move outside, and they mostly take turns. And we had squirrels jump from a rigid beam first, and then had them jump from a beam 3 times more compliant. And on the first jump that you see here, they undershoot badly and can't even contact with their hind legs. But by their 5th jump, they land with little error. And in over 100 jumps, not a single squirrel fell. So then we got clever and wanted to explore the sensory motor limits of jumping. So we moved the perch higher and further away. And with 3 high-speed cameras going, recording, and the students ready to see what they did, here's what the squirrel did. So it parkoured creatively off the wall to add a control point. And honestly, it looked at us as if, it was insulted because like it's a squirrel. But we'd never seen it behavior, it never did that before. So to understand how animals represent their environment, like we just saw, and make decisions, we were fortunately collaborating with Noah Cowan and Jin Kiram at Johns Hopkins, where they measure hippocampal place cells that signal a location in the environment. And I know Janelia is working in this area as well. They designed a dome with landmarks having an adjustable gap with the goal of measuring biomechanical decisions in real time, trying to do what we're doing with the squirrels outside. And they found that the hippocampus showed previews of the decisions during vicarious trial and error deliberation before crossing the ditch, differentially encoding literally jumping versus ditching. So you could tell what they were thinking about. And finally, here are some of the first attempts at using the principles learned that I talked about to make the most agile robots that someday can innovate. The robot on the top left is a robot with a spine. The one on the bottom shows jumping, both from Dan Kotichek's lab at the University of Pennsylvania. And the upper right is a monopod hopper designed by Justin Yim and my colleague, Ron Fearon. There's clearly a long way to go before we understand the fundamentals of decision learning and innovation. Thank you.
Great, Bob. Thank you very much. Wonderful talk. So there are a lot of fun comments about squirrel parkour coming in the chat box, and while others are typing in questions in the Q&A, let me ask you one, which is, so you showed that squirrels were within 5 trials able to learn a novel and learn how to adjust to a novel environment. What if other squirrels are watching that squirrel learn? Can they, just by association, just by watching, do they improve their performance in achieving that task?
Yes, we wondered about that, and it's not easy to set up that experiment in free-ranging squirrels outside. And what I would say is, maybe as you'd expect from individual variation, some seem very intent at looking at this and figuring it out, and others could care less. And so that we'd love to do that experiment in a more controlled setting, because there's so many different types of learning that we'd like to explore. We're beginning with Lucia Jacobs, actually a developmental trajectory. So learning over developmental time. So she's starting with little baby squirrels in a controlled environment where we're able to see what they do over time, and then we can actually look at performance in the same. So I think that the whole area of learning, especially at the limits of performance where critical decisions are required, I think is a grand challenge.
Great. Next question, Gwyneth.
Yeah, I guess following on from that, Bob, and really great talk. So you, in one part of your talk, mentioned embodied cognition, maybe with relation to the cockroach. And then you have the squirrels, kind of maybe doing cognition with their nervous system. But I guess I was wondering, in the squirrel scenario, is there any aspect of that computation that you think is more embodied?
Yeah, so I didn't get to talk about that because we have 10 minutes, but that's a major part of this myriad that we have. We're working to identify both the wonderful anisotropic properties of spines as well as legs and feet. And we work with an incredible metamaterials person at the University of Pennsylvania, Sue Yang, where they're creating, using kirigami and origami, along with liquid crystal elastomers, literal legs and feet and backbones that you can put collapse dimension properties in that you couldn't do with all 100 muscles and tendons, to really look at then the system level behavior of function. So that's a large part of what we're working on. And we think that the use of metamaterials in generating physical models, ultimately people are working on these to even replace components in people. But you could use these certainly, and will in 15 years, to replace out of parts in creatures to test hypotheses that you otherwise couldn't do, because you can vary things like cartilage and bone in ways that you couldn't really do otherwise. Thank you for that setup.
And then I think the last question, in the interest of time, it really builds on this theme, which is from Margaret. And the question is, do robots embody all the decision-making in the animals? Can you elaborate on the feedback process the field uses about learning these themes using biomechanics and iterating into robot design?
Yeah, that's one of the great questions. The answer is robots are so stupid, it's just tragic. And so we try to put a lot of their control into their bodies, but that's really the next great challenge. And I think that what has to happen is that we have to think about these decision makings of the animal. How does it know what its body can do? And how does it know that the environment will allow it to do it? And so what we try to do is test those very things in the robots with relatively limited sensory information, rapid information for high-level behaviors, and then see how they can succeed or not. So it is a fundamentally iterative process, and it's becoming easier as the sensors improve and as our ability to collect enormous amounts of data that we can correlate. I think we'll hear that later, maybe from Mackenzie, incredible amounts of data we can collect that we can be able to look at those correlations and then match them with the physics models, the multi-scale physics models, to begin to then have that realization of what can I do and what am I perceiving is possible. Because I have no idea, I watch the squirrels out in my backyard, how they know when they're being chased by a cat or something, that they can leap this far and land on a branch like that, and they don't miss. I mean, it's just phenomenal. So it'll be it's definitely a 15-year project to understand creativity and innovation. But yes, it's an iterative process with both mathematical and physical models.
Great, thank you so much. Thank you, Bob, for a great talk. The next speaker in this series is Sheila Patek from Duke University.
Hello, can everybody see my slides?
Yes.
Awesome. Thank you so much for the invitation to speak here today and participate in this workshop. It has been awesome. So awesome that at 9 o'clock last night, I was like processing yesterday's talks and wrote a new one. And what I decided to write a new talk on is this notion of merging pragmatic with audacious. We were told to be audacious today, but then, I was like, we need some pragmatic. So there's going to be talk about pragmaticism or pragmatic audaciousness from the lens of mechanics, variation, and integration. So I just want to start with audacious. I think like everybody who's been talking these 2 days, we do audacious experiments. And so just a quick lens of what the sort of large focus of my research program has been on, and then we'll switch over to pragmatically audacious, has been on this, this world of ultra fast, which is that we study systems that produce the equivalent of a movement lasting 1 second that takes 1 year to prepare. And this is essentially a problem of an extreme cascade of energy control, and it's the basis of ultra fast movements. And our primary study system is the mantis shrimp. And the reason behind this extreme cascade is basically it takes a very long time for a muscle to load energy into a spring. A latch releases that energy or controls the release of the energy from the spring. That turns into the motion of the system and impact and ultimately the most extraordinary power amplification you can pretty much get, which is the cavitation bubble collapse. So we, through work with the myriad team, that I lead, and I've been leading for 6 years now, we developed a class of systems, or named a class of systems called latch-mediated spring actuation. These are basically, movements and mechanisms that allow lightweight systems to achieve this extremely rapid energy flow to the environment and can be used repeatedly and repeatably. And this is an image of some of the systems we work on. But what I really wanted to point out here is the mass, range, and the acceleration. Here's the acceleration of a bullet in a gun. And this is the world that we're studying here, which is basically the left-hand upper limit to extreme acceleration movements that are not self-destructive in biology. And in the process of doing this, we worked very hard to come up with an interdisciplinary principle to why systems are able to do this or how they're able to do this. And ultimately what we found is that, or we were able to explain, is a mass transition that works both for engineering and biology, below which it works better to actuate with a spring and above which it's better to actuate with a motor. So, this is a lot of audaciousness because basically these systems, for us to even see them at all, or barely, we're filming at 300,000 frames per second. We can barely resolve what's happening here spatially and temporally, and almost nothing to be seen in terms of the energy flow through these materials that are actuating these movements. But the thing is, this is what happened last night, and I just kept thinking about, well, I enjoyed the entire day yesterday, but I was really nudged by two talks, Hannah and Rama's talks, that were approaches to multi-level and multidisciplinary principles of evolutionary diversification. Hannah proposed this synthetic connective piece, which is feedback loops and sensitivity to them, something that we could follow through all these levels of analysis. And I'm oversimplifying this. And Rama was talking more about statistics. And I kept thinking about that and loving it. And then coming back to a synthesis that Derek Briggs wrote a couple of years ago, he's a paleontologist, was capturing the history of this field, which Dan alluded to at the start of this workshop, which is the very foundation of biology and the history of biology and paleontology, is this problem of multi-level integration to understand evolution. So essentially what I decided to do is talk about, or, throw this to all of you to discuss, hopefully, how do we get from audacious experimentalism to audaciously pragmatic within the mighty paradigm of this workshop. So what I decided to do is pose a question here that I actually have been working on for about a decade with quite a few other people, which is, how do the laws of physics interface with evolutionary processes to yield integrated, multi-level, diverse systems? And all day yesterday, like Dan mentioned at the beginning of today, I kept hearing the same pieces of physics that we tackle at the whole organism biomechanics level as are being tackled at every level all the way down. So that propelled me to put out here, which I think we could all agree, all levels, analyses, and disciplines are operating within the laws of physics. And then there was a short discussion yesterday on this, but I'm going to say all levels in biological systems are operating within the foundational process of biology. And that is that variation is the engine of biological diversification. That is the food that is consumed to create diversity. So let's play with this a bit. So, okay, on the side, right, we've got these impossibly fast organisms, serious technical challenges that I would love Janelia to help me solve, and just simply visualizing energy flow through materials, for example. But at the same time, we can do something, and like the paleontologists have done historically, we just say, well, okay, what is one thing that we can measure that we know is real? And how do we piece this together to connect these levels of analysis and tell us something big about the history of life on the planet? So what I'm going to show you briefly is what happened when we picked a simple common mechanical system, which is levers or linkages. We focus on a basic interdisciplinary metric: size, rotation amplification, and sensitivity. I'll come back to sensitivity in a minute. That came up yesterday, actually. Test variation across levels, development, population, species, and clades. And I will talk about species and clades today, but I've worked on the other two. And finally, test the effects of size and variation on the pace, which is the rate and pattern, the transitions of evolutionary diversification. So if you're going to do something like this, it means sort of saying, oh, my experimental side that wants to dig and is currently and still digging into the intricacies of all the connections from molecules up to materials in motion, and instead to just pull out one thing that we can measure across these levels and capture this variation. And it was really through the genius of 3 postdocs who worked in my lab over the period of a decade, and they're all professors at these places now, that we boiled down this diversity into a multi-level problem with a shared physics of linkages. So, and as you'll see in a minute, we added in some other data sets from other scientists. The first thing that we found is this thing called 8-to-1 mapping that's been identified in many other studies of functional morphology. In a nutshell, what it shows is that you can get a lot of the same outputs from different configurations or different outputs from the same configurations across evolutionary diversity. This has been considered the engine for evolutionary diversification. I would be even more agnostic. I would say that this is just how diversity, evolutionary diversification, explores physics, and you get all kinds of cool outputs. What we did, though, is we went one level down and said, well, instead of just looking at the output, this is don't worry about the output, it's just a rotation amplification. And let's look at the links themselves. We expected to see this great ball of many-to-one mapping or mechanical equivalence. And instead, what we found is really something quite different. Some links, like the orange one, have very little association with the output. And then one link has a lot of influence on the output, the KT. This we term mechanical sensitivity, and probably every single person on this call today has performed a sensitivity analysis or knows what that is. It just means how much is the output of a system or an equation sensitive to a particular component. When we went and dug into that, and looked at it from an evolutionary perspective, we found that the components to which the output is most sensitive are evolving the fastest. Crazy, right? And anything's possible here. One level down, sensitivity, bigger level, higher evolutionary rate. So furthermore, when we looked at this across other data sets from other scientists, we found the same thing. The most tightly correlated components with the greatest sensitivity evolve the most quickly, and they're always the smallest. Oh, see, sensitivity and size, two things that I think we've talked about at least 10 times in the last 2 days. Newer work has shown totally different researchers, different system, the same thing, positive correlation between sensitivity and evolutionary rate. And then other researchers are looking at these questions all the way to the level of gene expression. What happens to a lever system and what's the variation around that during development? So I'm going to propose that the laws of physics interface with evolutionary processes through mechanical sensitivity to yield integrated multi-level diverse systems. And that this is a place that we can come together through our shared vision of whether it's a feedback loop or a sensitivity analysis or machine learning or forces or displacements, and see how they connect up across all these levels, and then find out how evolutionary processes tug on this space, this physics space of variation. And as a pragmatically audacious vision, and again, I didn't really come up with this. This is sort of the history of our field, but maybe notched up now that we have awesome phylogenetic trees, rates of change, abilities to measure these things across all levels, right? But basically simple metrics, simple mechanical system output, measure the variation across levels, test the mechanical sensitivity and redundancy, test the correlation with rates across levels, and the outcome is a multi-level process-based understanding of the mechanical pathways and principles of evolutionary diversification. I want to end on one sort of crucial interdisciplinary point. Biological systems rely on variation as the engine of diversification. However, engineering design is grounded in the reduction or the accommodation of variation. And I know so many people in this workshop work in this world, right? Either you're an engineer working on biology or a biologist working with engineers. And just as Bob just talked about, moving across robotics and biology with totally different notions of the utility and relevance of variation. And essentially what I'm proposing here is that variation is the crucial conceptual and experimental interdisciplinary intersection for these multi-level translational research areas. So with that, I hope I lit a fire on a few of you, and I'll be delighted to take some questions. Thank you.
Great, thanks Sheila for a very audacious talk and your energy. We've got some questions kind of being populated right now. I want to ask, maybe start off with a question of my own, which is, you know, when you look to like simple linkage systems, you know, how far out do you need to look to really be able to capture the dynamics of that system? And so specifically, I'm thinking here of Beth Brainerd's work, where they found in fish feeding linkage systems, like the epaxial muscle actually powers a lot of the suction feeding, whereas before people just concentrating on 4-bar linkages were just looking within the skull of the fish. So I don't know if you had thoughts on that.
Yeah, so one of the things that obviously in 10 minutes, it's like impossible to get under the hood here, but I have thought a lot about this over the past decade. So every system has its own story, right? Every system has its own details. But if we can pull just a little bit away from that and instead ask, how much is the system sensitive to one of its components? How much variation is happening in the individual components that contribute to the output? Then we start to have a more generalized framework in which to study the specifics of each of these systems. So this is why I got so excited at 9 o'clock last night, because I was like, oh, this is what we were getting at yesterday with like these feedback loops. Like, I just love the notion that negative feedback has to be everywhere, right? So what is the overarching thing that we can tug on that connects them together? And I think that this is that piece of like audacious experimentation that everybody here is so good at, right? Where we can get into the specifics of which muscle generates which output, and we should keep doing that. But right along in parallel, say, let me let go of that a little bit and ask, for a smaller component, does it vary proportionally the same amount? Or does a smaller component actually, just by virtue of its size, have greater variation? And that's why you would channel, or why evolutionary diversification rates would be enhanced there. So that's not a super satisfying answer, but that's kind of the lens that I'm trying to tug on, because I felt like we were kind of headed this way, or at least that's where my head went.
Awesome. We've got one quick question from Wyatt before we need to move on.
Yeah, so I mean, this may need to go into the discussion, but it's somewhat audacious. So if we should be using variation, I love the concept of looking at variation. What is your opinion about studying model organisms where some of that variation may not be present in the laboratory animals?
Yeah, I love it. So, that's a huge question, right? So have model organisms been selected to have less variation, or, given just even the laboratory environment? So this is, again, what I think. I think that the issue of ambient variation across levels is like super interesting, even in a highly sort of constrained system like generations of fruit flies or generations of mice, because then you can say, oh, look at that. There's the same variation, let's say, at the developmental level in both laboratory and wild systems. And then you can see where it gets reduced or funneled, or you may find that it's the same sensitivity axes, mechanical sensitivity axes, but the rate of variation across a population is reduced. So it creates a lot of different multi-level spaces where you can ask different types of questions with these.
Okay, in the interest of time, we're gonna move on. Next up to bat is Tom Daniel from the University of Washington. Tom, the stage is yours.
Thank you. I'll attempt my screen share as we speak. And is this looking good? Great, thank you. It's always a challenge coming on the heels of many outstanding talks, the two just preceding, making it even more challenging. So I want to thank Sheila and Bob for setting the stage. I'm going to continue in this theme, but I may dive back down into the single cellular level. And I just wanted to nod to a slight editing issue here, which is exploring how cells work in tissues and in whole organisms. And for my single cell colleagues, well, my colleagues who study single cells as whole organisms themselves. So I'm going to kind of give you a bit of an outline and explain who this is here. This is George Heilmeyer. He was, for better or for worse, a director of DARPA, and has various reputations, but I really like his catechism. It's called the Heilmeyer Catechism. It's the outline of my talk. And it begins with, what's your big idea? What is the state of the art? And the follow-on for that is what now, why now, and what is boiling out there in the world of science and technology? What's missing? And And in fact, people have been pulling a lot on that fourth thread, what is missing? And I think this is something I'm going to bring up just a couple of points there. And then I'd like to conclude with why would Janelia be uniquely positioned to pursue something in this space, okay? So I won't be as audacious or pragmatic as Sheila, but let me begin with the big idea. And the big idea we've been hearing for today and yesterday is these are multi-scale, dynamical, mechanochemical processes that involve multiple systems. And my particular focus is control and dynamics of movement. And just let me lay out a little bit about my lens here, and then I'll dive down into more detail. So with a focus on movement control, and hats off, by the way, to Gwyneth, Margaret, Wyatt, and Dan for creating such a moving workshop. It is very much in this sort of closed loop world we tend to think about things. There's some open loop aspects, but in fact, there's aspects of sensing, central neural system processing that Bob talked about, and others as well. There's motor output that programs information into muscles that dynamically generate propulsive forces and controls the motion of structures which produce forces, which change the sensory world. And this continues on and on and on. And I tend to look at it in one or a few organisms. This is the hawk moth Manduca sexta. This is something many of us study. I believe Simon is on board as well. And it does all of this. Fundamentally, paramecium senses. It doesn't have much of a central nervous system, but it has sort of motor output. Doesn't really have muscles, but has cilia. And those may change the world of paramecium experiences. And so for the moth, it experiences sensory information, centrally processed. The motor program comes out. That motor program drives muscle activity, which moves propulsive forces, which in fact represents a whole series of transformations. And I think everybody's alluded to this. I'm just wording it slightly differently. The physical world is transformed into a neurally relevant world through sensors. Mechanosensors are one example, where they transform forces and strains into neural signals that can be interpreted by the nervous system. In and of themselves, they are a multi-scale problem. That is, the structure, the skeleton, the interaction with the environment drives deformations of dendritic processes, which drives channel deformation, which drives calcium fluxes, which drives signaling. That in turn comes through another series of multi-layer processes that drive muscles, which, as you saw in the beautiful talks earlier today, that have themselves a deep and really interesting multi-scale problem, from the whole muscle to the sarcomere to the filaments to even the level of the single motor proteins. What we have said and what we've heard is that events at the sort of angstrom scale affect events at the nanometer scale, affect events at the micrometer scale, affect events at the centimeter and higher scales. And the interesting thing is the same is true in the other direction. This is a bidirectional feedback. I think we've heard this alluded to, and I'm going to raise some challenges about this in a bit. And some of the questions we might ask is, to what extent does the design of a motor protein influence the context of motion of an organism? And to what extent is the organism's interactions with the environment influencing the dynamics of the motor proteins, their likelihood of binding and force generation and the like? Their force sensing, as Malcolm Irving beautifully brought up earlier. So it is indeed a multi-scale system. For movement control, we can see that at single-celled organisms. There's a beautiful study underway, pioneered by Adrian Fairhall, coupling body mechanics, muscle mechanics, neural signaling, accounting for absolutely every neuron in a hydra. It's a beautiful animal. To the workhorse / fly of Janelia, to this gorgeous organism, and even to beating hearts. These are all problems of motion control. These movements are also associated with development, tumorigenesis, and even the creation and sort of maintenance of effectively organoids. So there's a brain organoid I just put up there, which I find really interesting. So the big idea is it's multi-scale. What's the state of the art? Why now? What's boiling right now? You saw examples over the last few days of stunning imaging technologies, time-resolved X-ray diffraction. I'll speak a little more about that. Myriad force and strain measurements at multiple scales, temporally and physically. Gene editing technology, stem cell and organoid tech, imaging across massive spatial scales. I have here something from a recent paper of ours of imaging of that Malcolm referred to, our time-resolved X-ray diffraction that allows you to resolve spacing of the myofilaments, as well as the dynamic stretch of the thick and thin filaments while an organism is performing a function. And in fact, Sage Mallingen, a recent graduate out of my lab, was able to record the electrical activity of the muscle, the spacing, the radial spacing of the filaments, and even the stretching of the thick filament as it's undergoing force generation. To her dismay, she found that you could not easily predict the spacing and stretching of the thick filament without knowledge of the other muscles being activated at the same time. So the total dynamics of the system was a confound for her. So there's a lot boiling. And in fact, in parallel, there's a massive explosion of AI, molecular dynamics, agent-based models. We've developed some. Ed Monroe referred to a beautiful series of agent-based models in the cytoskeleton. You see from Hannah El-Samad the importance of control theoretics of thinking, and also from Bob and Dan Kodachek's work. There's a series of hard inverse problems. And in a sense, what Sheila was referring to are these problems of saying, how sensitive is it to parameter variation? And how does parameter variation of one parameter depend on the state of all others? These are really, really fun problems, but there are inroads for them. So what's missing? And I sort of started to think about this, and over the last 2 days I've been struggling, but let me keep going. There's a lot going on. There's an incredible talent. And the thing that we're missing is sort of this scaling issue is actually, and bear with me, a principled way of coupling across scales. And I'll just go over a few of these now, but this is a unique moment where we can take computational advances, recent ones, and couple them with amazing, stunning data production and data acquisition and try and build up on this. So some of the things you may have already heard about are coarse graining. So you take, this model of 2 microseconds in the life of a myosin motor, 2 microseconds, and took, oh, by the way, 30 days of 1 GPU. So it's a bit impractical, okay? But you would definitely want to coarse grain that if you want to have hundreds of these interacting motor molecules in this beast here, in this myosin network. Let me go back a slide. And in fact, we've done some coarse graining recently using data-driven methods, and we're using data-driven methods largely from simulation. And there's the emergence of really exciting heterogeneous multi-scale methods developed by somebody with the coolest name at Princeton, Winan E. And that is not an abbreviation, that is the last name. These are powerful methods, and they do very well on going from the smaller scale to the bigger scale. It is much more challenging to do the opposite. And I'm pulling on a single paper that I found really interesting by Kathleen Champion. She was a graduate student in applied mathematics, working with Steve Brunton and Nathan Cutts at UW. And this is a direct quote from the paper. A major challenge in the study of dynamical systems is that of model discovery. That is taking real data of the sort that you're seeing coming through this and turning that into models that are not just predictive, but provide some insight into the nature of the underlying dynamical systems that generated those data. The parenthesis S is 9. And why I find this interesting is they raise several issues. Is how do you deal with this with dynamical systems of many simultaneous timescales, right? As was referenced by basically all the prior talks. And secondly is how do you optimize how you measure these systems across all those different scales? How can you inform yourself best? And I won't go into the gory details, but they point out that this problem is made challenging by the fact that many of the subsystems have different time scales, fast, slow. You could have way fast, fast, slow, super slow. You could have multiple time scales, and you can posit in a sort of control theoretic context, much as Hannah El-Samad would have done or has done, is there some dependence of the state variables, that is you, whatever, on the state of those variables or time rate, and then some coupling terms to other time scale variables, okay? And I'm going to gloss. It's a beautiful paper. I've given the reference at the bottom, but the basic idea is you take data to drive the discovery of these matrix coefficients that couple across scale. And in doing so, you learn about those connections, kind of like what Sheila was suggesting. It was this parameter, you know, this component of the matrix, right, which really drove the dependency. It had the highest weighting function across those scales. So I wanted to just bring that up as, for me, the major challenge is developing a data-driven computational approach to formally and principally couple across scales. So what would Janelia bring to the table? Why do this as a 15 and 15? That's 15 teams in 15 years. Well, first of all, it is intimately connected with the acquisition of data at the highest level. And that is something Janelia has been doing at the level of imaging, at the level of even whole organism behavior, cellular, molecular imaging, cryo-EM. The technology and the capacity to build technology at any scale at Janelia is definitely there, okay? Whether it's a human or a Drosophila, whatever your organism of choice is, I'm a little agnostic, okay? You're even welcome to do a moth. The second thing is if the teams are working simultaneously and you have computational teams working with them, just as we've done agent-based modeling and then developed deep neural networks to have reduced order models, much like Bob's template and anchor approach to higher spatial scales, right? So too can you do the reverse direction by this sort of more, it's called SINDY, but S-I-N-D-Y. The method's developed in the CUTS lab. So I have a feeling that a future effort should be one in which you are tightly coupling a formal method of coupling across scales, okay? And yeah, honestly, there's a lot of discussion about what system is optimal for that. I actually think it almost doesn't matter as long as you have that goal of that bidirectional coupling. That's the one that's been the one keeping me up at night.
Hey, Tom, we're almost out of time.
I'm done.
Oh, wonderful. Perfect timing.
And I'll just end with my favorite quote from fluid mechanics about coupling across scales. The world, this is not new in physics. This is not new to try and do this. This is a hard problem. And as was identified in 1922, big whirls have little whirls that feed on their velocity, and little whirls have lesser whirls, and so on to viscosity. I thank you all. And I will
Tom, thank you so much for that great talk. There are a number of questions that I think we're going to have to save for the discussion so that we can stay on time. And we'll now transition to our selected speaker, Jasmine Narodi from Rockefeller and the University of Oxford. Jasmine.
Let me share my screen. Is this clear?
Perfect.
Okay, great. All right. Hello, everybody. I want to start first by thanking the organizers for the chance to be part of this really incredible lineup, and in particular, this awesome session. So I'm going to talk today about some of my work on the motility of two organismal systems. So I'm going to split my time in between flagellated bacteria and moss-dwelling tardigrades. And these stories both nicely reflect, I think, the title of this session, and so integrative mechanics, as well as more broadly on several of the themes that have come up throughout this workshop across all the different sessions. And one thing that really struck me both today and yesterday, which Bob and Sheila and others have commented on, is that across all these scales and levels of organization that we've been discussing, the big questions that we ask overlap quite a bit. And one question that came up a few times, in particular yesterday, is this idea of ecology, of considering the performance of biological systems in the context of their natural environments. And this is really near and dear to my heart. It's very tied into the questions that I think about, which concern how biomechanical systems navigate natural environments. And these can be fluctuating and heterogeneous. So for instance, animals often have to traverse varied terrain, or on a longer time scale, they're dealing with changes in their environment due to seasonal flooding. And similarly, at the single cell level, organisms have to interact with complex environments like our intestines or soil, or with each other when they form these complex aggregates like biofilms or tissues. And finally, as we heard about yesterday, subcellular machines also are operating in really dense dynamic surroundings. And because it's not always possible to do all these measurements that we'd like to do out in the real world, even though I think we're getting closer and closer to that goal, one of the main challenges that comes out in my work is to try and understand what the naturally occurring mechanical challenges facing my system of interest are, so that any hypothesis or experimental design that I utilize takes these into account. And so the two systems that I said I would talk about kind of lie at opposite ends of the spectrum, but in line with what we've heard a lot about over the past 2 days, and that Tom focused on in particular, is that both of these stories are going to call for us to consider levels of organization above or below from where we start out. So at the end, we're kind of going to end up meeting in the middle. So first, I'll start over here with a molecular machine that drives swimming in flagellated bacteria, the bacterial flagellar motor. And this system was particularly appealing to me because across all this variation, like Sheila was talking about, there is a conserved core set of proteins. And I'm showing these here as solid colored regions. And these shared bits included the two components that are crucial for torque generation. And this is shown here in yellow, the stator, and the rotor C-ring shown here in red. And this was really exciting for me because it allows us to build off of experiments that have been done in well-developed model systems like E. coli, and towards this more ultimate goal of understanding this variation on this common theme. And when I first started building a model for this fundamental process, it helped to start me off to think of a macroscopic analogy. The structure of the motor lent itself really nicely to that of a playground merry-go-round. And the rotor here is ring-shaped. It looks like the merry-go-round itself. And the stator units, an E. coli motor can have anywhere between 1 to 11 of these units surrounding the rotor. These act as our friends that are pushing us along to entertain us. And like a merry-go-round, the motor can go at a huge range of speeds. This data on the E. coli motor's torque-speed relationship was collected by Howard Berg's lab at Harvard. And each of these points represents a measurement that was taken from an immobilized bacterium whose flagellar filament was sheared off and replaced by a bead. And by using beads of different sizes, we can get out a curve like this by calculating the torque that's generated by a motor to be able to rotate a bead of radius r at some observed rotational speed omega. And for me, from here, the obvious next step is to think about which parts of this range matter for motors in real bacteria. So these can spend portions of their life cycle in single-celled planktonic phases, swimming around, as well as in aggregates, like in biofilms or swarms. And what it turns out is that actually this entire range matters for real bacteria. So free-swimming bacteria mostly live out here in the low torque, high motor speed region because flagellar filaments are thin and they don't impart that much torque onto the motor. But when a cell comes into contact with very viscous fluids or with mucus or with other bacteria, these interactions then quickly shoot it over up here into the high torque region. And so what's salient here, additionally, in gut bacteria like E. coli, is that they're not only dealing with both of these environments, but they're often transitioning between them quite quickly. So this is something that their flagellar apparatus also has to be equipped to do. And if we want to characterize these kind of transitions in the lab, we have to think about how we're able to recreate this kind of dynamic environment. So with the standard bead assay that I described, we can test out a single point on the torque speed curve, depending on the bead size. And this is a single static measurement that doesn't tell us that much about how the motor allows bacteria to navigate complex landscapes. However, if instead of a single polystyrene bead, we use a superparamagnetic bead, we then can use an external rotating field to switch rapidly between different speeds in a single bacterium. And this allows us to test motor behavior across the entire functional range. And so for the sake of intuition and time, let's think a little bit about how we might deal with these kind of switches on the playground. So if our best friend wants a push, we're happy to take on the job ourselves. They don't make the merry-go-round too heavy and this is not too tiring. But then once everyone else starts seeing how much fun this is and they all start wanting to join in, it might get a bit tougher. And then you might want to recruit a little bit of help from some other kids. And it turns out that that's exactly what the flagellar motor does. So what we observed was that E. coli motors are dynamically remodeling themselves in response to changes in external load. So motors at high torque here maintain a higher number of engaged stator units than motors here at low load. And this dynamic behavior is what allows bacteria like E. coli to be able to move through and compete in complex landscapes throughout their life cycles. And if we were to ask these exact same questions, so how a biological system deals with mechanical challenges associated with the variability that arises in natural environments, but here at the other end of the spectrum, now we're no longer making measurements on subunit exchange or modifications in protein structure, but instead we're testing for behavioral changes as animals are encountering changes in their surroundings. And for this, we turn to tardigrades because they're an exciting system to this end because they're found in this wide range of varied environments, from marine to freshwater to limnoterrestrial. And they're also exciting for me mechanistically because they're one of the only soft-bodied animals with legs. And this allows for really nice characterization given that they have these discrete ground contacts. So using the setup shown here, we filmed our animals with a high-speed camera walking along an agarose shell from top and side views. And from this video, we found that tardigrades walked with this tetrapod-like pattern. And this is called as such because diagonal legs swing or lift off together in sets of 2, leaving 4 legs, a tetrapod, on the ground. And so we measured the relative timing between contralateral leg pairs, these are leg pairs on opposing sides of the body, and ipsilateral leg pairs, so directly anterior and posterior neighbors. And we found that across all walking speeds that we could measure, tardigrades maintain the same exact coordination with an ipsilateral phase offset of about 1/3 and an anti-phase contralateral offset, so just alternating contralateral legs. But in addition to having to walk at different speeds for different survival tests, tardigrades also have to face variations in substrate properties. So we wanted to see how they might be able to sense and react to changes in the stiffness of their ground substrate. And so we were able to vary the stiffness of the gel substrate they walked on. And what we found was upon sensing lower substrate stiffnesses, tardigrades maintained ipsilateral phase offsets, but switched to a kind of galloping movement with contralateral legs being in phase once the ground started to give away. And these patterns across speeds and substrates were reminiscent to us of those that were observed in insects. And in particular, we found that they matched nicely with stepping patterns that had been characterized in this recent massive Drosophila data set that's been collected. And now for us to be able to make any hypotheses about what underlying structures might be able to generate these patterns at the organismal level, we shifted our attention one level down. And the authors behind the fly dataset had proposed a simple circuit shown here, and a natural place for this circuit to exist would be in the ventral nerve cord, or the VNC. And this consists of 3 segmented ganglia, one for each pair of legs shown here on the left. And the general topology of the VNC is reasonably conserved across arthropods, as well as between arthropods and tardigrades. And this was really exciting to me in a similar way, calling back to the conserved core proteins in the flagellar motor. And so this is the note that I kind of want to end on, echoing this call for diversity in study systems beyond these standard models that Manu brought up yesterday and Sheila talked about today. And just like we can look to the E. coli motor as this jumping off point towards understanding these observed variations in flagellar structure, I'm really excited for this prospect that these recent deep dives into Drosophila walking might be a first step towards understanding non-model organisms in the panarthropod clade, and in particular to have these kind of more generalizable, simple models that then can be tweaked to think about this diversity of systems. Okay, so I will stop there now, and I just want to thank some of the places that have supported my work and people that I've been super lucky to work with our shared favorite organisms here.
Great, thank you, Jasmine. Thank you for hands down the cutest video we're ever going to see for this workshop. We have a couple minutes, so you can populate with questions. There's one question right now that says, over which timescales do the flagellar motors remodel as a response to load?
Yeah, so in seconds to minutes. So depending on how sudden how large the changes, they can do it within seconds, 5 to 10 seconds.
Another question is from Tom. It says, is it possible to image the neural activity simultaneously? They're pretty transparent looking.
That would be great. Yeah, we would love to do something like that. And I'm very excited to hear about possible techniques to be able to do something like that.
I had a question. You left with a beautiful slide showing kind of the diversity of tardigrades, and you talked about the ecology kind of in natural environments. And I guess it harkens back a little bit to a talk yesterday when I believe it was Dumont was talking about cell spindles and, referencing them as kind of large machines that have to be studied in vivo. You can't kind of reconstitute it in a petri dish. I guess what are your thoughts on that when you bring an organism into the lab, and try to study it? This is kind of more general question.
Yeah, and I think this is one of the things that is just constantly on my mind, and I brought it up a little bit at the beginning, right? The experiments that I tried to do, I tried to take into account what the organism might be feeling in its natural environment and the challenges that it might be dealing with. I try to recreate those, and so I like ideally to have some aspect of field measurements made so that I understand what exactly the organism is dealing with. And this is not always so feasible, right? With bacteria, this is really challenging to do kind of field measurements with them. Although I guess maybe Manu is more well versed in this kind of toolkit than I am. But with tardigrades, we were kind of aware that this particular species lives on moss, which is a complex substrate. So we knew that substrate stiffness might be something that we would be able to vary.
Great. I think with that, maybe we can transition into the discussion itself. You know, on behalf of Wyatt and I, we were kind of tickled to be involved in kind of such a great kind of array of talks. I think this, to me, no bias, but was very exciting because this is where we kind of really threw the doors open and started to really attack integrative biology at many different scales. So we had, you know, Sheila talking about integrating evolution, and Bob Folt talking about integrating mechanics with the brain, kind of cognitive biomechanics, integrating with the environment. Tom talking about multi-systems integration. And previously, the morning talks talked about kind of tissue, especially in cardiac muscle, and all of yesterday, we're looking at kind of cellular level. And there were just some parallels. I almost don't know where to kind of start, so I'll just kind of throw out, you know, kind of maybe the first thing and see if we can get the ball going with that. And it's this idea that Bob brought up with cognitive biomechanics, and it touches a little bit on what Tom was saying, that kind of biomechanics is boiling over now. It's boiling over into different fields. And you know, how much of you know, biomechanics is going to intersect with the brain? I think we're seeing that kind of quite a bit now. And this idea yesterday that was brought up of kind of feed forward sensing. So how you can be predictive. I think squirrels are showing us that you can do that. And I was wondering if there's any parallels at the small scale and the large organismal scale. So I guess I wanted to start off with that, so you'll have questions on that. I see a hand up from Simon.
Sorry, I raised my hand before you posed that question. So we should go after that question first, because I had a question that was sort of very different.
Okay. We'll see. I'll give a second to see. Brains don't exist without bodies, says Manu.
Yeah, I mean, I just find that that it has taken so long for the community to accept this, but it's ironic in so many ways. I mean, here everybody would agree, but we have spent so much time in neural systems completely ignoring the biomechanics aspect of it.
Point well taken. Kiisa, did you have a related question before I go over?
Yeah, mine is definitely related. So I just want to say that, you know, we all have to assume, I mean, this is kind of deeply philosophical, but we all have to assume that the future is going to resemble the past, but we can't predict exactly what's going to happen. And it's the intrinsic dynamics of the muscles that manages that for us. So one of the things that the brain has to learn about the body is what that is, to enough of an extent to not fall out of the tree.
I think it's a point well taken. If there's no other kind of questions on that, we can do.
I think Bob has a response.
Bob has.
Bob. Okay, Bob, your squirrels.
Yeah, the most recent work we've done, which we haven't completed, suggests that Manu and Kiisa are right on target, which is we've now put them through a difficult course whereby they had many choices of which branch to jump on, each with different challenges. And the preliminary results suggest that if it's a relatively simple biomechanical task that is not really pushing the neuromechanics and musculoskeletal system, they just take a very simple solution. But if you make it really difficult, that is push them to the absolute limits of what they can do, you also don't see a lot of innovation and creativity. But that middle area is where they're willing to explore and create and always confound our experiments every time with things we never thought of. And so I think that that it is the link to the body and the biomechanics that really puts the decision in a position where they're assessing what they can do and they're making their best guess about the environment. And I don't know how they do this, because we built plexiglass panels so they wouldn't interact with the posts that we had for the branches or the bars that we had. And it looked impossible. That's why we made them. And so, sure enough, when we made the branch too hard to jump on, they just jumped in the air and landed on top of the plexiglass sheet with no problem at all. How did they know that their body could do that? And how did they know the thing wouldn't just break? But they never fell, right? It's incredible. So I think it's very exciting integration, just like you're talking about, that you can't forget about the biomechanics as you look at the creativity and innovation aspect.
So maybe continuing on, it's the theme of brains. Ed Monroe, do you have a comment on that?
Yeah, I just wanted to echo something that Manu said, but from the perspective of cell and developmental biology. I think we've spent a lot of time thinking about systems for signaling and transcriptional control in cell and developmental biology without thinking at all about how those systems are controlling architectures and mechanics. And I think that we're starting to realize that a lot of those, the signaling systems are essentially erected upon architectural scaffolds that they're busy controlling, and that their assembly dynamics and activities are contingent upon the thing that they assemble themselves on. So I think it's, in some ways, it's early days for cell and developmental biology, but it's fundamentally the same kind of questions that we need to address that the people have been talking about all day today.
Great, thanks for that, Ed. Manu, you had a comment?
Yeah, I think I just wanted everybody to, something that Tom has brought up, this idea of model discovery. I think we have not talked enough about that in this group. I think Tom gave one example. I know, for example, there is a recent work in active matter from Jorn Dunkel's lab, given a bunch of fields, which could be chemical concentrations of a whole bunch of molecules, polymer, actin, myosin, but you don't know who's turning anything on and off, but these are spatiotemporal fields. They were able to apply machine learning to come up with some plausible models. Tom, I think I would love to hear from your perspective on, I think the challenge that I find is, even with that sort of an approach with so many possibilities, and again, it's not clear to me whether that approach would eventually, in some clear systems, it will be merged with the bottom up. But when so much is unknown in the middle, I get worried sometimes. So maybe people could reflect, and especially people who work on machine learning and discovery could comment on this. I know it's the wave of the future, but I think, where does the human creativity versus really let machine discover these models? Where do you all land on that?
I think Hannah has a related comment.
Maybe if I can just do a quick response to Manu, which is, thank you, I agree. Model discovery, it's an emerging space. I mean, I think that field is still in its infancy. There's a lot boiling right there. What I find interesting about it is, in fact, if it's coupled and sort of girded in by appropriate physics, okay, it's not free to, in Sheila's words, not free to violate those principles. I think model discovery with data-driven approaches is good. There's a recent paper also by Krithika Monohar and others, and I'll try and get the link to that. She also does model discovery. These are all in physical systems. So to everybody here, I'm unaware of this in living systems, but Hannah may have a better knowledge of that.
Hannah?
So actually, Manu, there is a very similar set of ideas that actually that people are using in identifying models from, for example, sequencing data. For example, there is a new paper by Jonathan Weizmann's lab where they look at they do some really smart labeling to look at RNA, different RNAs, and they identify the vector field, like the trajectory of RNA, messenger RNA from different genes on a genomic level. And then they use deep neural networks to infer models out of that. They do a combination of dynamical modeling and deep neural networks. And I think these kind of approaches, even though they're just for genomic data, and I don't think they're thinking beyond that, and some elaborations thereof will be very important moving forward. I think there was a mention yesterday of machine learning and how it can be constrained by previous knowledge. And that's true. I think you can derive physical constraints on, the neural networks or whatever based on, on previous knowledge. But I don't think that's sufficient, actually. I think we have to go deeper than that. The prior knowledge cannot just be, boundary conditions. It has to be integrated in more meaningful ways. And how to do that, I think, is the cutting edge of this.
Great, thanks, Hannah.
Jimmy, that led into my question. Yep. So we came.
Awesome. I saw you nodding. So take us away, Simon.
Tom, did you want to respond to that first before I went on?
Couldn't agree more with Hannah again. Absolutely. I just want to say that we are recently developing DNNs, neural networks that are hypersparse. And when you sparsify, you reveal connections between parameters you had not anticipated. And I think that's sort of fun. But I'll leave it at that.
So the thing that I wanted to bring up was along these lines, and I'm very, when we've talked about these elusive general principles that we've tried to identify before, and I think we heard some really nice examples actually from all the talks. I've seen many of those elements before, I think actually from all four of you, so I sort of knew what was coming. But I really like it, like we're seeing Gates in Jasmine's talk, the leg stiffness in Bob's talk, the mechanisms in Sheila's talk for power exchange, muscle mechanisms and force transmission that we heard earlier. Tom brought up these arrows. And Bob brought these arrows going from anchored to template models or from higher scales to lower scales. And I think oftentimes we've talked about our general principles of sort of identifying the templates that are common across a lot of systems where you might have the same complexity reducing to the same model. This comes up to elements that we talked about yesterday, Manu, right? And I think a big challenge going forward is, Bob's called these posture principles before, they're reduction principles or multi-scale mechanisms. There's a lot of different terms for those. And I think, Tom, but correct me if I'm wrong, that the modeling framework that you were talking about and what you were just talking about, Hannah, was to try to identify those things that bring us across scales. So if we identify that in one system, we have a template, we have an animal, we study a beautiful tardigrade or a cockroach or a moth or a stomatopod, and we then show how that arrow works and we build up a model that's in that case, and they have a DNN underlying it or a sparse DNN underlying it. How do we generalize that? And I think, how do we say that there's a general principle across that arrow so that the reduction is common? And what are the features of that? And that's where I think it's challenging, and I want to see a path going forward, because a lot of what we're doing to do that, I think, as a field is turning to ML methods, DNN methods, but those are very hard to interpret. So it's very hard to say, as my DNN that brings my anchoring from a complex system to this template in one system, similar to the thing that would do it in this system. So what's the path forward to that? Is it integrating TDEs into that and making them more physics-based DNNs and sort of trying to work towards convergence? Or can we find non-dimensional numbers? Can we find a Reynolds number about the transitions that go across? What's the path forward in that space?
Great question. I will make a rough attempt at this. I think that is the question, Simon. So one of the things that you can note is that when you couple deterministic models that themselves have, gazillions of parameters, and the parameters can vary, when you build DNNs on those, right? And by the way, those models are connected to reality, like data, right? When you build DNNs on those, it is less of a black box than the traditional DNNs of here are 1,000 faces. Can I classify this? It's not a classification when you have a handle on the underlying mechanisms and how the DNN represents that. So that's a very vague answer, and that's about as deep as my brain has gotten into this. We've been certainly doing this problem, using DNNs for inverse problems, saying what were the parameters that best compensate for deficits or do X or do Y. But once you have guardrails of data and physics, right? That's, I think, a better solution, better way forward. But I defer to wiser souls here, because this is a new area for me.
Just to push it further, I think that helps, but it doesn't get us all the way there yet, right? Because there's still a large part of that's very data-driven. And that data-driven part is always going to be hard to generalize across systems when we get that element. At least it sounds like it. Maybe there's paths forward.
Hannah, you have your hand up?
Yeah, I mean, I'm actually trying to understand the question. So if the question is methodological in the sense that, let's say I come up with a way to coarse grain, backward and forward, how can we generalize for my system? How can we generalize it to your system? I think this is a tractable, potentially a tractable methodological problem. If your question is, are we going to coarse grain, 10 systems and come up with equations of motions of biology? I'm not sure this is, I'm not sure that there are equations of motions of all of biology, much like, there are, So I think we should formulate, a set of problems that are tractable. And I'm not sure there's a Reynolds number or whatever of all of biology, right? But maybe I'm not understanding your question correctly.
Yeah, I certainly wasn't meaning all of biology. I was meaning if you have similar kinds of systems that are reducing down in the same way, or where you have the same, where the common multi-scale directionality. And I think actually the controls is the way to get to it. I really liked what you were talking about with controls. We've done it at the organismal level and manipulating control loops and sort of seeing that, because then you can sort of say, okay, even if this system isn't just feedback linearization, it acts like that. And that allows for a simplification of this higher dimensional dynamical system, or the scale, this degree of complexity at a lower level that gives convergence at a higher level, or emergence at a higher level. Even. And I want to find systems that do that in the same way, not to just come to the same point, so that we don't just get the same gates out of it. Because, for example, in Jasmine, in your gate thing, right, like the tardigrades don't have to worry too much about gravity. So it's actually interesting that they get some of the same rates of systems that do. And in the gravitational systems, even flies probably don't have to deal with them. In the gravitational systems, we think about Froude number. That's why I was thinking about that as sort of governing gate transition. That doesn't probably apply at a tardigrade level, yet you see similar gate emergence. So what is the thing that's commonality that's leading to a gate convergence? I think you were touching on it. I'd actually push beyond flies even and sort of say, how does it relate to cockroaches, where it starts to matter, horses?
Right. Yeah. So the thing that kind of came out to us here is this common control structure in the VNC across arthropods, and the similar topologies in tardigrades. And this we did, without any kind of ML or any kind of neural network. This was just a complete hypothesis. And I think this is the kind of thing that maybe you were talking about, which is the commonality that has to exist within systems that you can kind of integrate in this way, that you can coarse grain in similar ways. And so speaking just quickly as a side note, the reason that we think that tardigrades care about this, even though that they don't necessarily care about gravity, is because falling over and stability matter still a little bit, because it makes them slower, it makes them less progressive. And so they care slightly about these things, maybe less than cockroaches would.
Thank you. I want to build on what Sheila was talking about. What is a pragmatically audacious vision that Janelia could participate in over the next 15 years? Is it this? Should we try to go all in on ML efforts to find these common principles? Should we go to model systems where we can study in great detail variation of like actuators, sensors, or, you know, should we go kind of very broad? So what's the titration of depth versus breadth? Jasmine, did you have your hand up?
Yes.
Great.
Yes. So I'm biased, but I am really drawn to systems where there is this kind of commonality between and then variations on a theme that lead to this huge diversity. And then the thing that I like about these kind of systems is that it allows for both breadth and depth, right? So in the flagellar motor, you have this common core set of proteins, so you can understand kind of build a model for how these core set proteins are interacting with each other to generate some mechanical performance metric set. And then you can look at the diversity around that has evolved in different environments. And you can ask so many questions about what the role between the environment and those individual parts are, which bits, you know, are more what affects performance more, what's the sensitivity of various parameters, what matters more at the organismal level, how do you control those parameters at the structural level, and there's just kind of, it's just such a rich set of questions in systems like that. And they exist at all scales.
Thank you so much. And thank you to all the participants and the panelists and the great questions. I think Janine, we're gonna take a very quick break now.
Yeah, let's do about 5-minute break. And we will resume around 1:55 Eastern.
Janine, how live are we right now? Is it just the panelists?
No, we are live to everybody.
Okay.
But you guys are welcome to chat, continue your discussion, anything you like, but it's live.
Yeah, could I actually ask Sheila a question?
Go for it, yeah.
Yeah, so I was wondering what your thoughts were on systems that are kind of mechanosensitive or flexible phenotypically, so non-genetic variation. So similar to how the flagellar motor, you know, remodels itself in different environments, how that kind of non-genetic variability might affect the rates of evolution, and kind of in the context of your the idea of sensitivity.
That's a really cool idea. You know, one of the things about this topic that makes some people love it and some people hate it is that it's an old one, right? And people have tackled this in so many different ways. And, you know, whether you're talking about like G by E or phenotypic plasticity, it still matters a very great deal. And I think that, you know, if I think these days, in short, the ability to develop variation in different environments is its own level of analysis. And that can be, you know, what Bob's talking about cognitively, or it could be literal morphological changes over time in a given environment or with different amounts of exercise. And these are the things where, you know, it's just so incredibly rarely done, where we would look at the capacity for variation at each one of these levels, including things like the, I mean, like you talked about with loading of these motors, that in and of itself is a relevant source of variation for selection, not necessarily that particular morphology is passed down, but the ability to respond in that way is passed along. So it's just, you know, it might not be glamorous, but it sure is hard to do and requires a lot of different types of scientists coming in and establishing these shared experiments across these levels and environments. And so, you know, when I came up with this to talk about last night, it was really me just like hearing so many of the same shared threads yesterday, and of course today as well, and just wishing to find this thing that we could all come to the table and measure in this, you know, and what we were told to do, right? The 15-year, 15 lab business. And I just kept coming back to that. But I think you're totally right. And I think it's just fascinating to think about that level of variation as well.
Yeah,
Sheila,
I also had a bit of a scramble last night after yesterday's talks and redid everything.
Hey, Ed.
Yeah. I have a question, sort of for Sheila, but for everybody really. Like, how much do you think can be learned, not by comparing variation across different organisms, but by inducing variation in particular experimental systems and looking how variation is coupled across different scales in the same system, like exploring the variational tendencies of specific, even well-studied experimental systems across multiple scales? I mean, is that another way to generate the kind of, the spectrum of variation that that Rama was talking about yesterday? It's a statistical ensemble of variation within an individual organism.
I really love that. And, I think some people are tired of this topic, but, there absolutely has to be tunable variation. I mean, we know, like, even among human populations, the fingers have different amounts of variation in length. Yeah. And if the pinky varied, absolute magnitude wise, the same amount as like the longer fingers, it would be really weird, right? So there's tunability relative to size in the variation of structures. And that's well known, like David Wake's work on like toes and development, right? And so the idea of being able to probe the scalability or the control of the scaling of variation through experimental systems, again, is an old question, but here we are in a space where that one thing would have taken a generation of work. And now we have these model systems where we can probe them
and the tools to induce the variation.
Yeah, like what is the physics of size, scaling, and variation in these systems? So I really like that.
I have to say, I love Ed and Sheila. I love this idea. And I think it's the convergence of, synthetic biology with that could be very powerful in the sense that we have not used actually engineering of biology to ask any multi-scale problems in any rigorous ways. And I think that could be very, very powerful.
Yeah, and I also really like this, and this was in your talk, Hannah, which I just absolutely loved, but the fact that you have this overlay of how a mechanical system's output is disproportionately sensitive to some components or another. And again, that could be in a feedback loop, or it could be in these other types of like universals, and why they would be concentrated in one piece of the system versus another. Why would a system be disproportionately sensitive to one component or another? And again, that's a pathway for evolutionary change. It's a pathway for control, but it leads straight to synthetic systems because it allows you to strategically design an integrated system where it makes sense why you would have one piece be changeable and one piece you want to have within that tolerance at all costs. So you start to get into like integrated synthetic systems with strategic variability built in, hence the universal of testing sensitivity analyses. I mean, we already know that that's important, but really different lenses on it when you're talking about synthetic versus biological. So yeah.
I'd like to give Rama a chance to make his comments, and then we're going to get started with the next session. So go ahead, Rama.
I was going to say just one really quick thing. It's really a placeholder for later maybe, but there is actually a really interesting fundamental difference between doing statistics over ensembles versus doing targeted variation of an individual solution and using the data to infer. There's actually a really interesting fundamental difference of those two things. Maybe later when we talk, we can discuss what that fundamental.
Yeah, I'd like to hear more about that.
I'd be happy to talk about that.
Yeah, I would love it because as I hope you saw, Rama, I was like, boom, my head exploded. And I was like, different way of thinking. They're very exciting. I'm looking forward to it.
It's really interesting because there's a really formal mathematical difference between the two things, but they relate to each other in an interesting way. We can get into it later on, perhaps. Thank you.
All right, thanks, guys. That was a great break time conversation. Let's move on to session 7, which is going to be moderated by Simon Sponberg from Georgia Tech and Matt McHenry from UC Irvine. So over to you guys.
Okay, great. So we're going to move on to thinking more about the comparative mechanics across scales with a little bit more focus on the environment and what's going on there this time. And our first speaker is going to be Andy Bewiner from Harvard, who's going to talk to us about all kinds of fun organisms, get us back to elements of muscle and connect that to organismal locomotion. So Andy, go ahead and take it away when you're ready.
Okay, thanks, Simon. Let me just get my screen up there. Okay, does everybody see my first slide here?
Looks good.
Okay, so thanks again. As many of us, I think, appreciate the organizers putting together this workshop. It really has been fascinating and a stimulating discussion, and really a coverage from a scale of mechanics at the subcellular now to whole animal biomechanics. And it's really nice to see biomechanics coming back to the fore alongside neuroscience. And so that's sort of the focus and theme that I'm going to focus on, which is sort of taking a neuromechanics approach to the integrative biomechanics of movement and motor control. And we start out at the millimeter scale, looking at muscle properties by being able to do muscle ergometry and measure force length and force velocity. And I know while Kiisa is critical of some of these classical measurements, they do inform how we can interpret the muscle dynamics in vivo. And we also do with work experiments, but these play into understanding how muscles interact with tendons, or muscles act on the skeleton to control the forces and the work that's generated, but potential for tendon energy savings relative to the timing of neuromuscular activation. And we do this by making measurements both during steady movement as well as non-steady movement. And we study both terrestrial locomotion. So in terms of environment, we're interested in terrestrial movement and the role of muscle tendon dynamics in relation to moving over solid substrates versus animals that fly through the air. We focus on the flight muscles of birds. And these muscle tendon dynamics then build up into limb dynamics, where we can record the ground reaction forces, at least in terrestrial systems, quantify joint torques and relate those to the movement, and whether we can ask the question of whether muscles and the muscles acting at particular joints act as motors to feed energy into the movement of the organism. Do they act as struts and allow for transfer of energy within the limb, or the possibility of elastic energy recovery from tendons? And or do they act as brakes to dissipate energy and help control and stabilize movement? And ultimately, control and stable, robust movement, whether it's flying through the air or running over the ground, requires a steady control of whole body dynamics. And that's controlling the torques about the center of mass as well as the energy state of the center of mass. And in relation to that, we have had collaborations with engineering roboticists in terms of building the Big Dog quadruped robot, where we use the lessons we learned from whole body and limb dynamics to try to inform the design of the robot. So very much like what Bob was talking about with Rex, you can use the engineering of the robot to test whether your dynamics of observed in the general principles, such as spring mass mechanics, apply to the controllability of your robot as it's moving over uneven terrain. So I'd like to focus on the in vivo measurements of muscle function in the context of bird flight, and then also compare that with terrestrial locomotion. So this is a cockatiel flying in our wind tunnel, and we can make measurements of the force development by the pectoralis muscle through time in relation to its length change by sonomicrometry, and relate that to the neuromuscular activation of the muscle as a bird flies over a course of a series of wing beat cycles at different flight speeds in the wind tunnel. And what we find is that the muscle generates force, not surprisingly, during the downstroke. This is the major motor for flight. And it does this while shortening over 35% of the muscle's length. So the muscle, as a result, does a considerable amount of work. So it activates very quickly, develops a high force as it's shortening, and that results in this counterclockwise in vivo work loop. So the muscle acts like a motor to produce the energy necessary to power flight. And when we can multiply these cycle patterns of work against wing beat frequency, we can calculate actually the mechanical power output of produced by the pectoralis muscle. And this gives us an insight into testing aerodynamic models for how power output varies as a function of flight speed, because we can measure that directly within this one large muscle that amounts to as much as 22% of the bird's body mass as it is trained to fly in the wind tunnel across a range of flight speeds. So in flight, the pectoralis muscle is designed to function as a motor, generating forces while it shortens over a considerable length range. In comparison, we looked measurements of animals running over ground. In this case, this is a guinea fowl running over an obstacle, so we call it an obstacle treadmill. And in this case, we're measuring muscle tendon forces from a force transducer mounted on the tendon. Again, sotomicrometry measures muscle length changes, and EMG for activation patterns. And what you're seeing is the synchronized recording from a guinea fowl. This is work that Monica Daly did in my lab when she was a PhD student, comparing the obstacle stride in the solid traces relative to the normal level running in the dash traces. So above is the length change pattern of the gastrocnemius muscle, and relative to its force pattern. And you can see that when the animal goes over the obstacle stride, the muscle doesn't shorten as much, its velocity of shortening is reduced. And so there's both a force length and a force velocity intrinsic stabilizing effect to increase the onset of force development and the magnitude of force development and work that's performed that allows for an intrinsic feedback control to stabilize and generate additional work, allowing time for neural reflexes to come into play. So to test the relative importance of reflexes, in this case, the intrinsic properties of the muscle being arguing to be very important, we actually followed up this study with a bilateral reinnervation study of the guinea fowl, again, of the lateral gastrocnemius. And when you section the nerve to the muscle and allow it to grow back, it regains its motor innervation. So the motor neuron motor units are reestablished, but the synaptic efficacy of the 1A spindle or the 1B Golgi feedback of length and force to the muscle is lost. So the muscle loses its proprioceptive feedback. And 13 weeks post reinnervation, as the motor recovery of these muscles has occurred, here's a guinea fowl running on a treadmill, showing no signs of suffering from the loss of its proprioceptive feedback to its muscles. And when we actually look at the function of the LG and look at the work loop patterns performed during strides preceding an obstacle step, going over the obstacle step, and then the stride beyond the obstacle step, the reinnervated LG in blue compared to control actually matches very much the same pattern we saw when the LG had its proprioceptive feedback intact. So in general, there's a relatively qualitatively very similar work loop pattern, largely based on intrinsic mechanics of the muscle. It's largely independent of LG proprioceptive feedback and control. So I'd like to turn to studies we've done with hummingbirds hovering in a virtual surround cylinder. This was inspired by previous work looking at hovering flight within insects. And so here's a hummingbird, ruby-throated hummingbird, hovering near a feeder, tracking the vertical surround, which you see in the lower left-hand panel of the video. This was from a rear projected surround that was rotated at different velocities. And what we found was when we tracked the motion of the head and the body of the hummingbird, we found similar to insects, hummingbirds track the motion of the surround with their head orientation in the center of gaze remarkably well. So it matches very much the pattern of the rate of the rotating surround, whether it's rotating in one direction or the other and different rates of rotation. And so there are occasional head saccades to reestablish a new gaze direction, but when it's tracking the surround, the head orientation precisely matches the rotational velocity of the surround. The body orientation lags behind the head orientation, but it actually follows along and basically follows the orientation of the head during free flight. Now, the reason I mention this is because this was stimulated by previous work from the Dickinson lab, in which they would tether flies on a magnetic support, allowing the fly to rotate and control yaw-based rotations relative to the surround, rotational surround, where the animal stabilizes gaze and negates optic flow as a result of that. And one of the real advantages, and this is the reason I switched over to visual motor control, is that the work in Janelia and in the Dickinson lab has very much taken advantage of the fly using flight as a behavior, that they can look at the integration of visual stimuli relative to motor output in terms of flight. So I'm going to highlight 3 examples using the fly as a really powerful neurogenetic behavioral model for linking nervous system activation dynamics relative to motor behavior output. So the first was a study from Michael Dickinson's lab, in which they did patch clamp recordings on a visual interneuron while the fly was behaving and flying and responding to these virtual surround optical flow cues. And they found that this neuron doubled its activation during flight. So it's basically sending descending activation of the flight motor system to turn on the fly's flight relative to when the fly was inactive pre-flight or post-flight. More recently, in collaboration with others, they've done 2-photon imaging of the ventral nerve cord of flies, which allows them to sample different populations of descending neurons from the ventral nerve cord associated with various motor behaviors of the fly, grooming versus walking in different directions or turning. And they're able to map different cell populations, identified cells based on calcium signaling imaging of these populations linked to these different motor behaviors. So they're beginning to sample within the population of neurons which ones control different specific motor behaviors. And finally, a very recent study that was in collaboration with Gwyneth and Wyatt and others at Janelia with others with taking advantage of really sophisticated optogenetic split GAL4 driver lines. They're able to study many flies. I can't remember how many, 64 or so, involving hundreds of descending motor neurons, which resulted in 1 million plus images of movement patterns of these flies. And they were able to then do machine learning or automatic tracking and a categorization of different flight behaviors. So they're able to create a 2D behavioral space for the fly mapped onto the different populations of descending neurons that they were able to record from by optogenetic calcium imaging. I will note though that the patterns of these behaviors in the 2D behavioral space, or in this case where the flies are undergoing different patterns of behavior for grooming and movement, are really largely qualitative and not really quantitative biomechanical measures of motor performance. So the caveats I would put out for the power of these imaging methods are really strong, but they really still lack the ability to get really good quantitative measures as we often can with certain muscles and certain larger organisms in the vertebrate realm. So that motor behavior is largely a qualitative measurement of activity, and it really doesn't involve the limb and body dynamics, the forces and torques and powers necessary to control body and limb motion. The other thing is that calcium imaging, which is also going to be done in transgenic mice, is largely restricted to anesthetized mice. They're not really freely behaving. And then recently, again, in a mouse, anesthetized mouse model, Huang et al. Showed that if you record electrophysiologically from specific neurons in which you are also looking at calcium imaging, that the spike measurements by the calcium imaging doesn't really capture a large proportion of the spikes that are recorded electrophysiologically. So there's got to be some caution taken into interpreting the level of activation based on calcium imaging. So with regard to the challenges and questions that Janelia would like to put out for us to try to address, I return to my questions because Gwyneth made a plea for this. So I'd like to ask the question, how can whole animal behavior in the context of the environment be assessed by integrating multiple systems? And I think we've heard this from several speakers, nervous, muscle, and skeletal across multiple scales of organization. And how do the underlying properties and collective interactions of cells within these systems contribute to emergent higher level function and ultimately the behavior of the whole organism? So the challenges to answering these questions, I think, and this is more or less an iteration on points that people have made in previous sessions in the earlier talks, is that developing methods for functional measurements of neural activity, and this is particularly the case for vertebrate animals, linked to a quantitative assessment of muscle function, biomechanics, and behaving animals is a real challenge. And on the alternative is that where you can make measurements of the biomechanical performance, we often don't have the mechanism for being able to link these clearly to the activity of neural control of sensory motor integration that would control these movements. And this certainly applies to more generally for flight and swimming, which are more challenging locomotor modes for being able to measure biomechanical performance directly in the context of neural activation and control. So what can Janelia do to be transformative and enabling, in my view? It's great at building new tools. It can contribute micro and nano electronics to record neural activity in behaving animals at the level of muscle function and neural activation in terms of muscle EMG, fiber strain and force. And developing technologies for recording neuromuscular parameters. For example, a recent paper reported a flexible viscoelastic hydrogel electrode that can match the tissue properties in which it's embedded so that it deforms as the tissue does, and it gives a more reliable recording of the electrical activity of that tissue. and also developing machine learning approaches for automated quantitative kinematics and natural motor behaviors, interface to inverse dynamics techniques. So it's really critical that these automated techniques for quantifying motion be interfaced to inverse dynamics techniques. And I think this is something that Mackenzie will certainly talk about in greater detail in her talk following mine. And then finally, as many have talked about, the importance of being able to create interdisciplinary collaborative teams of engineers and physicists that can interact with biologists and computational or AI scientists to tackle these really highly integrative and complex problems across scales. So with that, I'd be happy to take questions and just point out that over 2 different generations of my lab over time, most of the work that I've talked about today was done very much by the graduate students and postdocs that I've had the pleasure of working with over the years. So thanks.
All right, thanks, Andy. We're already a little over time, so we're gonna have to hold questions for the discussion section, and Matt will introduce our next speaker.
Yeah, thanks, Simon. So next up, we have Mackenzie Mathis from EPFL. And as soon as she has her slides up, she can get going.
Okay, let me see. I think Andrew's slides are still up, so it says I can't share mine.
I'm trying to get mine.
No worries.
There it is, okay.
Yeah. All right, let me try this again. All right, can you guys see my slides okay?
Yeah.
Perfect, great. Yeah, thank you so much to the organizers. This has been a fantastic day and a half or so of talks, and quite refreshing because I guess I don't know many of you, and so this is kind of a new crowd for me, and so I'm really excited to tell you a little bit about our work today. And yeah, it's been really inspiring. So my laboratory in general is really interested in understanding how the brain drives behavior. So we're certainly at the multicellular organism level, learn that single cells are very important. And in large part, we build tools to understand how we can actually measure this movement and relate it to neural activity. So in general, we kind of convert, as many of you do, combine neuroscience, engineering principles, and of course, a lot of machine vision and AI, which I'll mostly focus on today. And in general, our kind of larger mission is really to uncover foundational principles of neural computations in order to build more adaptive AI systems. So our typical model systems are actually either mice or neural networks, and we're very much interested in things like adaptability, robustness, and generalizability principles in both natural and artificial intelligence. But a large part of this has really been about actually being able to measure behavior, as I'm sure a lot of you sympathize with. Over the years, it's been challenging, even though there's been incredible examples. For example, what Andrew just showed, even with the marker guinea fowl, this is just incredibly beautiful. Or the hummingbirds, I have to ask you offline how you got the markers on the birds, but really impressive work that you're doing. And as you guys all know, that measuring movement has a rich and long history. So going back to Edward Muybridge and his zoopraxiscope, which was this beautiful kind of racetrack along with a series of camera traps in order to concatenate images to essentially make a video, to actually ask a question that was rooted in a bit of biomechanics, namely, does the horse lift all 4 of its feet off the ground when it's cantering? And nowadays we have incredible high-speed cameras that can reveal to things that would otherwise we couldn't see by eye, like this beautiful tap dancing dance from the bird while it's courting behavior. And there's been a lot of advances in terms of not only things that we've heard of quite a bit, like force plates and EMGs and marker-based motion tracking. So a really beautiful example from recent years from Grégoire Courtine and Sylvestre Messera at EPFL. And there's been a lot of incredible tech development on making flexible arrays for both EMG and like neurocognition type of studies. So here's an example from Stephanie LaCour's lab also at EPFL. And now I'm going to skip over kind of an entire field of computer science and just say that we've really been standing on the shoulders of giants in recent years and kind of, taking principles from deep learning and computer science in general to build new tools to say, how can we kind of capture motion from a completely non-invasive way as best as we possibly can. And so over the last few years, we've been working on this toolkit that we call Deep Lab Cut, which allows for efficient markerless pose estimation. And the efficiency here is that you can really get away with very little labeled data. So just to put it in perspective, these large scale sort of human data sets that people have built for computer science and computer vision benchmarks or often tens of thousands of individuals and, you know, up to 40,000 images, where that's not really scalable for the individual lab or even across potentially a model organism. And so we leveraged a technique called transfer learning, which allows us to take networks that have seen actually millions of images. So it's kind of imbued with very good starting weights, and then allow the user to fine tune it in a completely user definable way. So for example, I'm really interested in sensory motor control and adaptability across learning. And so one of our model organisms is mice, and we study the hand. And so of course, it's sort of possible to put markers on them, but they don't really like it. And especially to get the digits, which are very, very small, it's nearly impossible. And so what you're seeing on the right is an image of a mouse that wasn't in the original training data set. This is frame by frame. There's no post hoc filtering or anything. And our error is well within the width of the digit for this type of tracking. And so since this is the right crowd for this, I felt like maybe pointing out that it doesn't have to just be at the level of a mouse. It can be across many different things. So here's an example of using deep lab cut at the cellular level, where you might notice that it's one tracking all the cells in this dish, but it's also being used as the classification. So when the cell lyses, the color of the dot changes. And so that's actually indicating a change state. So you can really think about using key points in interesting ways. This has also been used in tissues. So for example, in medical applications for looking at the movement, or even tendon in a walking individual with an ultrasound. And of course, I'm mostly focused at the organism scale. Here's a nice example from data from Guping Feng's lab at MIT, where they're looking at socially interacting marmosets. And so over the last 3 years, it's been a bit of a whirlwind to make this toolbox open source, free and usable, and really trying to support the communities. We're really thankful to Chan Zuckerberg Initiative for funding the maintenance of this code base. And we just hit 230 downloads in the last 2.5 years. So it's been quite fun to see the uptake of the software and all the incredible applications. For this, I also just wanted to point out there's a nice paper, not from our group, but showing that actually that deep lab cut based tracking can be as accurate as X-ray based, like bead implementation. So I think for, the hardcore biomechanists in the room, often motion capture, as you know, like from the surface has this soft tissue artifact issue, but it can be used if you label the data in an accurate way, as accurate essentially as human labeling, which is really the key take home for that. Okay, but the question is what do you do with all this data? And maybe you had a question hopefully in mind when you started to collect it. And I'm not going to talk about all the different ways. We recently just published a review on, using machine learning algorithms in order to sort of analyze the outputs of such pose estimation toolkits. But I'd like to just focus in the next few slides on some of the more biomechanical focused work that we and others are doing. So as I mentioned, we're really interested in sensory motor control. So here's a couple examples, some unpublished work from our lab, but from Oli Keynes that looked at brainstem circuits, also from Sylvia Arbor's group using deep lab cut to again dissect brainstem circuits, and a really nice example from Janelia from Adam Huntman's lab, where they also use deep lab cut to look at reaching kinematics and do some really elegant work dissecting the role of thalamocortical interactions. Recently, just maybe as a bit of a case study, we've also been interested in teaming up with people studying more in the wild type of applications. So here was a paper that was just published at ICRA, where they're also interested in tail biomechanics. So I loved Bob's work is just amazing, but this is just kind of a fun application of, cheetahs don't particularly like wearing costumes. And so it was really fun to team up with Amir Patel's group at the University of Cape Town, South Africa, to collect some of these data sets. If you're interested, all of this data is completely open source. We labeled a very large scale data set so people can use it as a benchmark with a lot of 3D ground truth data as well. So we're very, very eager to see what people do with this type of work. Maybe just as a shameless advertisement, we're really interested in building machine learning tools, particularly for kinematic and biomechanical applications. So we'll have a new toolbox coming out soon called Kinematic. If you want to follow for updates, of course, we're on Twitter. But in the last few minutes, what I really wanted to do is kind of give you a vision of where we're going with this ourselves. And hopefully this inspires conversations amongst you as well. So we're very much interested in, say, taking measurements from the systems, like the biological agent, building artificial systems that mimic this as best we can. Of course, all models are wrong, some are useful. And then essentially reverse engineering and seeing where the model breaks when we perturb the biological agent or the artificial agent. And so a large part of this actually is taking motion capture data, fitting it to biomechanical models, solving the inverse kinematics that then essentially, inform these artificial agents. And so I'm sure you guys are familiar with this principle, but in general, what we do in the mice or in these artificial agents is we aim to make measurements of the functioning system and then perturb it. And this is a continual loop. So what type of agents or biological systems do I actually mean? So to give you an example for sensory motor control, and this has been touched on a few times, proprioception is a super interesting system. And so we've taken an approach called task-driven modeling that's been really well popularized, especially in vision by Jim DiCarlo and Don Yeomans and others, to try to say, like, how can we build models that sort of faithfully recapitulate the proprioceptive system as best we can? So I obviously don't have a lot of time today. There's a preprint on this work if you're interested, or happy to talk about it later. But long story short, getting proprioceptive data is really hard. And so what we did is take handwritten human characters, build inverse kinematic models in order to generate 1 million different trajectories in 3D space of reaching. And then we train these neural networks end to end in order to solve tasks like character decoding or character recognition, maybe what we think of as a higher order proprioceptive task. If you look at the level of the spindles, and so of course we have spindle models that underlie this, just with simple, say, T-SNE embeddings, you can see this is a really entangled input. But suffice it to say, with lots of the different models that we tested, these spatial temporal convolutional neural networks were quite interesting. You see, once they're learned, they start to detangle this proprioceptive information along the hierarchy. And then if you do sort of classical cellular physiology on these models, you see that they have tuning curves, much like have been reported in the primate cortex. You can also do things like generalized linear models to sort of, do unit classification of these neurons in the artificial neural network. And they also seem to have many of the same tuning properties that we see in biological systems. So lastly, what we're now working on essentially is building, solving the inverse kinematics and doing this in the mouse. We didn't have a mouse model, so it's been a few years where we've been working on such a full biomechanical model of the mouse. And then we essentially do sim to real. We extract then, the generated synthetic muscle activity and then compare it to real neurons in the brain. So just a few short take homes. Hopefully in this really short time period, I've convinced you that at least deep learning has really revolutionized our ability to analyze behavior, precision, and scale. I'm very excited about new approaches to modeling the sensory motor system. We think that, of course, these platforms are not only interested in just fitting data, but actually generating new hypotheses to test them. And lastly, I just put up this Humboldtian graph that maybe many of you know from Views of Nature. It just really struck me over the last day about like the scales and the ecology and the environment of the animal. I'm thinking about this in a much deeper and broader perspective. And I was always really inspired by Alexander von Humboldt for this. So with that, I added a little squirrel video just for Bob at the end, but thank you all for your attention and your time, and looking forward to the discussion that ensues.
Great, thank you so much, Mackenzie. We've got a few minutes for questions, and while hands raise and people type into the box, I'll just say that folks like Tom, Jimmy, and Manu were raving about Deep Lab Cut. And I include myself as a fan. Maybe I could pose a quick question for you, which is, if Janelia were to make a contribution to machine learning in your field, where do you think large contributions could be made?
Yeah, I mean, Janelia has already been really trailblazing in this space, right? Like Kristen Branson has built incredible tools from a neuroethology perspective. And so I imagine that they will continue to work in this space as well. And so I think there's a ton of synergies, right? Like, Deep Lab Cut is one of the pose estimation toolkits, and we're really glad that they use it and include it in their larger package. But there's a lot of things to do. So I think, from hearing themes across here today, it does sound like machine learning could potentially be a really interesting way to probe like across scales and in ways that haven't been necessarily connected outside Janelia. So it could be fun to see happen.
Jimmy, you have a question.
Yeah, I was wondering, what are your thoughts on, moving away from the visual kind of modality in terms of tracking animals and going to something like sound? What are the limitations and the challenges?
Yeah, it's a great question. So there are, of course, deep learning based approaches for doing things like speech recognition or a lot of natural language processing. There's wave nets to process sound information. Typically, the limitation is about localization, right? Like we're pretty good at this, but not that neural networks are so good at knowing like who's speaking when they're speaking. But certainly I think what I'm quite excited about are like integrated technologies. So using like sound, vision, RFID chips, GPS, EMG tracking, like all of these things can be combined to actually make models that take into account multiple modes of this type of data, right? And I think that we could potentially learn new principles about the latent factors that sort of drive behavior, and certainly from a biomechanics to a whole organism decision-making perspective.
Great. Sorry, Jimmy, but Dan's got one last question.
One quick question. Yeah, Mackenzie, beautiful stuff.
Thanks, Dan.
Yeah, and as I watched this and used Deep Lab Cut, and I was reminded from Andy's talk that, and I'm often reminded when we're using Deep Lab Cut that, and it fits in with the previous question, that kinematics are great and important, but sometimes we want to know about dynamics, and in particular dynamics and interaction with the environment, like ground reaction forces.
Yeah.
Do you see, ground reaction forces are presently measured in hard ground, and if you're lucky in flight or swimming using backing out techniques. What do you see in a kind of science fiction world where I can view an organism and all the forces it's applying in and on its environment? Is that possible, or is that silly?
Yeah, no, it's a fantastic question. And I think, so we certainly work on ways to say like just from motion predict EMG, right? So we can collect down, the same way that you use supervised machine learning or semi-supervised machine learning approaches. So that's possible. I would say ground forces are really hard though, right? Actually, I think there's still a lot of ways to go. Maybe anecdotally, like we know, for example, in the motion picture industry, this is one major limitation, is to like make it look real enough when people touch the ground using these type of approaches. And so I think this is not a solved problem. But one thing we're actually very much interested in my lab is actually like animal object or animal environment interactions. And so we do use a lot of 3D computer vision in order to say like reconstruct the entire world of the animal and see how they interact in that space. But I think ground reaction forces is a long ways off from being perfect. But yeah, we'll see.
That's on my personal wish list. Environment.
Yeah, no, that'd be happy to talk offline about it if you want.
Thanks so much, Mackenzie. There are a couple of great questions in the chat box, and I'll hand things back over to Simon.
Great. And so for our last talk of the day, we're going to have, or for the session, sorry, not of the day, we're going to have Christopher Pierce. Chris is here at Georgia Tech, and he's going to give our third talk. Go ahead, Chris.
So hopefully you can all see and hear me. All right. So first of all, I'd like to thank the organizers for giving me the opportunity to speak at what's been a really enlightening talk. Today, I'll be talking about how environmental forces can explain undulator neuromechanics in a couple of different systems. So we've seen some really beautiful examples of locomotion today in a lot of different contexts, and I don't think it's a tough sell to say it's important in a lot of aspects of organismal fitness, et cetera. And we know that locomotion in general is made of a multi-scale interaction between an organism and its environment. And to echo a point that was made in the earlier session, I'd like to really underline and focus on today that the environment isn't a small perturbation on what an organism would otherwise be doing, but in certain instances, a totally inseparable part of understanding the neuromechanics. And to get at that idea, we're going to talk in particular about a class of locomotors, namely lateral undulation. And this is a locomotor strategy that you can find in a lot of different physical environments, from the aquatic to the terradynamic and various other situations, and in a lot of different systems. Lateral undulation is achieved by a propagating wave of body bending that generally moves from head to the tail in a linear or largely linear organism. And that wave of body curvature is initiated by a traveling wave of muscle activation. And what we now know is that in general, that wave of muscle activation and that wave of traveling curvature need not occur at the same speed. They don't need to travel at the same speed. And in general, the phase relationship between the wave of traveling muscle activation and the wave of body curvature is known as the neuromechanical phase lag, which is thought to be a common feature of the lateral undulation template. This was sort of first encountered in some papers in the late 80s and 90s in a variety of aquatic systems. And since that time, the Goldman lab has actually extended the list of organisms where we can measure these things by considering a pterodynamic situation. And in particular, looking at the sandfish swimming lizard. What you see here in the bottom left of my screen is an X-ray video of a sandfish burrowing within sand. And this organism has been connected with electrodes at various points along its body, which allows a comparison of the muscle activation patterns and the body curvature traveling waves. So in addition to kind of an expanding range of animals where we can see neuromechanical phase lags, since their initial discovery, there's been several models advanced to try to explain the origin of this phenomena, including several bottom-up sort of style approaches. I'm highlighting one from the Holmes lab here, but there are others. And alternatively, the Goldman lab in this 2013 paper advanced a much, in some ways, simpler top-down model that really takes advantage of the fact that in the case of sandfish swimming, we're in a highly damped regime where there's no gliding, which makes it easier to calculate the body reaction forces of the environment, the reaction forces of the environment on the body. And it allows an explicit calculation of the torque that's required to realize a particular target kinematics. So kind of implicit in this model is this idea that the organism is targeting a particular sequence of shapes and it has a control goal to create some kind of sine-like motion that's going to get it through the environment. So using resistive force theory and some stuff that's known about the interaction of slender bodies with sand, you can input the kinematics, the assumed target kinematics, and then using the interactions with the forces in the environment, you can explicitly calculate the torques and how they move along the body. So you can then do this and directly compare this model with experiment by looking at the data that's collected, which is the form of body curvature as a function of time at particular segments. Here I'm looking at one particular segment towards the tail of the organism and comparing that with the onset of muscle activation. This is summarized nicely in this video where you can see the kinematics of the organism crawling through, and those patches that are lighting up are showing you when the muscle at that particular point is activated. Now to compare this with a theory, we have a nice graphical way of representing that with this space-time diagram, which allows us to keep track of the relative phases of these traveling waves of curvature, torque, and muscle activation by looking at where these wave fronts are moving at a particular point on the organism at a particular point in time. So for instance, this green curve predicts where the granular forces would conspire to create the necessary torques to realize the target motion. And you can see by looking at these data points that the onset of muscle activation and the offset of muscle activation agrees very well with the prediction of where the torques need to be to create that movement. So it turns out this trick works with other dissipative regimes, including what we'll focus on today, which is low Reynolds number swimming. And you'll notice there's a lot of organisms that swim in this dissipative regime, including the nematode C. elegans. And you can immediately see that the kinematics are fairly similar. And there's a slightly different reaction force because the environment has different properties than the granular media, but you can still produce this force versus velocity curve that allows you to compute torques. And this has been done in a variety of contexts where you can look at different muscle torque onset for different environments. So given this fact, we want to look at and say, can we move from electrodes to optics and study neuromechanical phase lags in the much simpler system? Are they even there? And then can we capitalize on what tools we already have for C. elegans to measure neuromechanical phase lags? So how do we do this? We have a strain which was engineered by Bill Schaefer's lab that expresses a red fluorescent protein and a green calcium sensitive protein, GCAMP. The red fluorescent protein allows us to account for motion artifacts that would otherwise obscure the calcium transients. And glossing over a lot of details, we have to align the two images. We have to extract the edge and the center line of the organism. We can then chop it up into discrete segments and calculate the ratio of the green and the red and these different segments in either the positive or negative side of the organism and compare that to the curvature. So how does this look dynamically in the case of swimming? Well, we have our swimmer here, and you can look at the curvature chymograph, which has these nice alternating bands of positive and negative curvature. And for both sides of the organism, we construct these bands of positive of muscle activation and relaxation. And the big question we want to ask is, do these waves travel at the same speed? It's kind of hard to see that with the chymograms because of noise and other artifacts. So it's a little easier to see this by looking at some line cuts. So in this case, I've chopped the organism into 9 segments, mathematically speaking, and I can look at the curvature oscillations in blue, the positive side muscle contractions, and the negative side, muscle activations in yellow. And by looking at how these waves move along the body, you can see very clearly that the muscle waves are moving faster than the curvature waves in the context of swimming. So if we plot that up as a phase lag, we can compare it to our theoretical prediction. So here I'm reminding you of the sandfish comparison of torque onset with muscle onset in the model and in the experiment. In the middle panel, I'm showing you all these different environments where we can do this trick of calculating explicitly where the torques need to fall. And then for the low Reynolds number swimming context, I'm comparing the onset of the muscle from the phase lag we calculated to the theory. And we have a very surprisingly good fit given the very large differences between these organisms, which really underlines this idea that similar environments can produce similar dynamics across systems. C. elegans is known to adapt its gait to a variety of different environments. And today I'll just talk about comparing swimming in buffer to crawling on agar, which is their most common culturing environment. And in agar, you can see that the motion is generally slower and the wavelengths are shorter. And also we expect the interaction with the surrounding medium to be dramatically different from the buffer. So if we do the same trick, we can actually see that in this context, the muscles and the curvature waves are more or less synchronous with some small details swept aside. And overall, the wave speeds travel at the same speed. So if we put that back into our phase plot, you can see that we're getting a good agreement with a damping model of environmental interaction forces, where things are more synchronous. So really the point I want to make today is that environment can unite organisms at very different scales and from very different taxa, but also for a particular organism, the environment is a strong perturbation that can change the underlying neuromechanics. So that's all well and good, but what about complex environments? We do tend to think of C. elegans as somewhat of a lab animal. It's been cultured on plates for decades and certainly isn't representative of other more exotic nematodes in the environment, but it was isolated from a complex environment. And that environment involves dense, viscoelastic, heterogeneous environments. And from what little information we have about the ecology of C. elegans and other nematodes, we know that from their very small perspective, they have to undergo kind of a fantastic voyage in the course of their life cycle. They operate in boom or bust cycles. When they run out of food and this rotting vegetable matter, they have to find their way to another source of bacteria. And they do that by traversing large-scale barren soil environments, so sort of wet granular environments. And they often have an ability to hitch a ride on neighboring insects to get them from place to place. So from the standpoint of environmental interactions, that's a lot of diversity that they have to contend with. So I'll finally end with a quick outlook. How can we approach environmental realism in neuromechanics in the case of C. elegans? Well, one thing we've been looking at that I don't have time to go to in detail is placing worms in viscoelastic gels. And in this environment, we in general see much more complex aperiodic motion in contrasted with agar crawling or swimming. We've also looked at the more well-studied environment of microfluidic host arrays. And what we'd like to do is not just image kinematics in these more exotic proxies of environmental conditions, but do functional imaging and continue to ratchet up the complexity of the terrain in the lab, inspired by the environment. Next, one of the nice things about C. elegans is there's sort of an embarrassment of riches in terms of how much information we have about lower level neural circuits. And there have been numerous models advanced. I'm highlighting one from the Cohen group. It integrates a lot of neurobiological information into a complex bottom-up model. we'd like to reconcile that with these simpler top-down models that were developed more on the organismal biology side. And finally, we want to tie these things together by evaluating emerging hypotheses with robophysical models. Some of my colleagues in the Goldman lab have been developing undulating snake or nematode-inspired actuators that have more passive mechanical properties akin to a natural undulator than their non-compliant counterparts. And we can see in this preliminary experiment that with a very simple open control loop and a little bit of passive compliance, more like a nematode, you can actually navigate through an obstacle. So I'm over time, so I'll end there and just thank my mentors and my funding and everyone for your attention. Thanks.
Great, thank you, Chris. That's excellent. We'll go to just really quick questions. Dan, you have your hand up. Did you have a question for Chris, or is that from before?
That's from before, sorry.
Okay, I'll ask a question. So you said that the similar kinds of neuromechanical phase lags or neuromechanical phase lag in general is emerging in several different organisms interacting with pretty different environments, although they all have some similar elements to them. And I was wondering if maybe you could touch on the question I asked earlier, what is the commonality that's leading to that similar emergence across the systems?
I mean, I think it's just, in some ways, it's sort of a simple mechanical fact that the torques you need to produce a undulating motion. And remember, not every undulating pattern is necessarily an efficient one kinematically. So it's just a simple, you kind of have to do, just sort of stare at these body reaction force videos, and it's not necessarily intuitive, but it's just that there's this conspiracy of the mechanics that says, if you want waveform X, you have to apply torques there. And I imagine that that's what the organisms through iteration are targeting. It's certainly probably a little more complicated in the case of high Reynolds number swimming, where you don't have as simple of an environmental interaction. And perhaps the inertia of the organism is more at play. But in the surprisingly large array of dissipative undulators, which is a lot of different organisms, you can kind of use this trick and have a model that's relatively straightforward to compare to experiment that has some degree of mechanical intuition behind it.
Okay, great. So I think we move on to the discussion section now. Is that right, Janine? Yeah.
Yep, it's discussion time.
And Matt will start us off.
Okay, thanks, Simon. Well, first, I have a question for Andy in particular, although anybody could chime in. And Andy, you had highlighted muscle and neurophysiology as exciting areas where perhaps the 4DCP program could make contributions. And I know that you have thought a lot about connective tissue, and bone and cartilage and tendons. And I wonder if the program might be well poised to make contributions to our understanding in terms of, along the theme of vertical integration and mechanics at multiple levels of organization. I wonder if connective tissues might be an interesting area for a contribution as well.
So I think that's a really good point, Matt. Given the length of time, covering muscle function in relation to nervous system function was obviously difficult to do in a 10-minute talk. But clearly, the forces that muscles generate and transmit through tendons, so one is connective tissue of tendon or ligaments, that's important, but those are the forces that get transmitted to the skeleton. And we heard an earlier talk today about proprioceptive loss and how that can have negative impacts on the skeleton. So the interaction of muscle forces, which are probably the largest forces that skeletal elements see, both in the invertebrate as well as the vertebrate realm. And so those forces probably play an important role on skeletal development and skeletal maintenance. And so we know that bone loss and bone remodeling are very much influenced by changing dynamics of the forces and the opposed stress and strain that the bone cells see. So I think a broader approach to thinking about the interaction of different systems, so not only skeletal, but also cartilage, could be an important broader concept for integrating how these different systems interact at different hierarchical levels of organization.
I had a couple of questions related to experimental systems that might make sense for 4DCP. So it would seem to me that given that 4DCP has this interest in cellular physiology in vivo, in the organism, that would lend itself to optical approaches and therefore small organisms that are transparent, like the tardigrades and the nematodes. And I wonder for those among the speakers who study larger organisms, what might be lost by focusing on small critters?
Well, I would say we now know that small critters and invertebrates are not necessarily simple. They may be more amenable to the kinds of experimental measurements that one can make, and you don't have IQs looking over your shoulder as to what you're doing to the poor critter. But they're definitely not simple. And as somebody pointed out earlier, the control of muscle function in invertebrates is complicated by the fact that they're inhibitory motor neurons as well as excitatory ones, different from vertebrate systems. So I think the paradigm for how motor control gets played out in invertebrate systems is in part reflects relatively fewer numbers of motor neurons, but a more sophisticated variety of ways in which muscles can be activated and controlled. So coming back to the point of larger organisms, I think the challenge of larger organisms is, one, can you have facilities that you can actually house them and study them? It's possible, though, to put transducers in larger organisms that it's difficult to do for small organisms. That was one of my points, is if you can develop smaller devices that can be embedded in the smaller organism, then you can take advantage of the fact that you can do studies that are much more manageable on a small invertebrate or a small vertebrate organism. So I don't know that I would say that I think that the advantage of the large organisms is basically implantable devices that you can then make recordings from under in vivo conditions. And I don't think we have, outside of, as I said, the mouse system allows for optical methods for measuring cellular activity of the nervous system, but it's generally from anesthetized preparations. They're not freely behaving animals. For that work, you have to end up using these electrophysiological tetrode arrays to make those kinds of measurements.
That's true, but I mean, it has changed quite a bit. Like, I think most of the mouse papers are now certainly awake and behaving in the last 5 or 6 years for all the calcium studies, for example. Like, just as an aside, Andy, but I mean, I agree. And coming back to the question, yeah, I don't think any of these things are a this or that, right? I think the animal kingdom is fascinating, so I don't think it has to be a play against each other. I think as humans, we like to just categorize and pit things, but I think that it's sort of a short-sighted thing, right? Like, every organism is interesting and it developed in its own ecological niche, so why don't you study it? Many people feel like some model organisms are closer to the model of a human, but I mean, a mouse is not a human, much like a nematode is not a human, right? So for me, I think it's equally interesting to study these systems and hopefully do comparatives, across these different systems is where we hopefully work towards, right? Like, we want principles of neuroscience. We don't want the textbook to just keep getting bigger. So,
yeah, I mean, I reinforce that point is I think the comparative approach broadly across different organisms and size and modes of locomotion, it really will inform us in terms of what are the guiding organizational principles by which mechanics gets integrated with movement control.
Absolutely. I agree.
Yeah, if I could just chime in quickly. I mean, I don't really have an answer to that question, but I can offer a question I've thought a lot about in conjunction with the neuromechanical phase lags, which is if you kind of buy this premise that there's common physics that is kind of allowing some commonality of the neuromechanics in this very broad top-down model. If you look at the sort of anchoring details in a nematode versus a fish or a lizard, it's interesting to wonder, is there anything you could learn from the much simpler structure of the nematode in terms of the broad principles of neurological organization at some coarse grain scale in the larger animal? Or on the other hand, are they coming at the same emergent neuromechanics from a completely useless to make the comparison completely different way? That's something I've been thinking about.
Yeah, maybe as the last point in this, like, I mean, you can always best answer it from your own perspective, right? So for me, I actually really like mice because you can teach them to do so much and you can really probe learning on very short time scales. So for me, to understand dynamics is to understand the process of learning. And so that's why I like kind of like, bigger organisms in this sense. It's just they can do more. Like, let's just bluntly, right? Like they can just do a bit more. So.
Okay, Jimmy, you have a question.
Yeah, my question is kind of touches on all the speakers, and it's basically like my dream would be to measure stuff in wild animals, right? And the reason is because that's where it's most relevant. So I don't have to worry about, am I getting it right? Am I approximating it right? That's actually what they do. You know, I swim fish all day long in my flow tank. I'm guessing at what they're doing mostly, right? And I'm telling a story and I'm publishing a paper, but the fish is swimming like much slower than I ever swim it in the flow tank. Then what am I really addressing, right? So I was wondering what people thought of advancing the techniques to be able to do these recordings on truly wild animals and interfacing that with the fields of movement ecology, as well as like fishery science. Like there's just other adjacent fields that would benefit from this type of information.
Well, I had thought about including large-scale movement and free-range movement patterns of animals in the realm of, I mean, movement ecology is really about the ecology of how movement intersects with ecological factors influencing organismal behavior. But just simply quantifying, as you were saying, animal performance in a field setting, what are animals really doing in the field? And that's, you know, like work that Alan Wilson's done and others are doing, where they are able to get GPS mapped onto accelerometry and magnetometry data to really understand what the dynamics of movement are, you know, predator-prey relationships or how fish are swimming in the field. Those are really important directions to go. And I think we're, with sort of small scale electronics and the improved computational ability, I mean, we're essentially right now trying to build a little Arduino heart rate recorder to fly birds around and measure their heart rate during voluntary flight rather than having them in a wind tunnel to see what kind of, that tells us about the heart rate function in relation to flight behavior. So those tools are becoming increasingly available, easy to program, and I think that's definitely a direction. And Janelia could certainly move in that direction if they really wanted to move out to monitoring, you know, performance of animals in a field natural environment setting.
Vanna, you had a question.
I think I had a comment on the, I think connecting both to what Jimmy said, but also what was previously discussed. There's something unique about a certain class of systems, and we all choose them for specific reasons. For example, in invertebrates, they undergo metamorphosis while having behavior. And interestingly, we have discussed motility, but motility is both coming from cilia and muscles. And in these systems, they're controlling via neuromuscular control both of these systems at the same time. Not only that, they're actually transforming their morphology while they're still encoding and controlling behavior in a period of weeks to months. So again, going back to, it's always relevant in terms of questions, but again, you get to watch certain sets of things that when you choose, and many of the same principles that are being described in the context of control of muscle dynamics, if you really go back to the evolution of the neuromuscular junction, there are lots of philosophical ideas around that cilia control was the first goal for neuronal systems. There is a lot to learn from there and then actually apply it back in a systems manner. So again, I think choice of system is really either will be doomed or it's very, very careful, very important.
I think that's an excellent point, and it raises like a good reminder that we need to be flexible, right? Like we shouldn't always limit ourselves to what we know. Like being a scientist means that we should be uncomfortable with the unknown, right? Like if your question isn't good for it, then go somewhere else. Yeah, so I like this idea of the morphological and the changes. We've actually been playing around with using like self-supervised learning approaches to teach artificial agents that are undergoing morphological changes. So it'd be really fun. I don't like connect with any biologists that look at this question, but if there's someone has an interest in this, I'd be really happy to chat with them. Maybe not in the last 5 minutes of this, but offline would be great. So very cool, yeah.
Great. I'm going to hand things over to Simon. There are, Simon, a number of raised hands, but I'll let you take over.
Yeah, Stuart, you had your hand up, so we can go to you next. And then I might pose a question before we move to Dan.
Okay, so this, I mean, it was already partially brought up a little bit, but to throw learning back into the mix, in terms of things we talked about previously, both evolution and physics constraining things, but then Mackenzie, you had brought it up a little bit, but I was reminded of other videos, YouTube videos I've seen of squirrels and the way they go through obstacle mazes. And there's a huge diversity, like each squirrel has a personality essentially that has to have been shaped by its experiences in the environment and the way that it's learned to approach problems. And thinking of that both as a interesting thing to study, but also a problematic thing to study. And sort of I'm curious what people's perspective is on whether or not that's something we should stay away from because it's just much too difficult, or whether that's the most interesting thing. And that also brings up some organisms, some big animals are much better at learning and adapting than small ones. And so that could be, a complementary sort of comparison for differences in learning.
Well, yeah, go ahead, please, Andy. Yeah.
Well, I was just going to mention that we all have personalities and we approach different situations and tasks in different ways. And I think we all, this comes back to Sheila's point of biology is in part driven by variability. That's how diversification evolves and arises. And so I think we by ignoring variability across individuals and across different kinds of motor behaviors, we're losing insight into and how personality can drive that. In fact, Ben de Bijvoort has shown that flies of similar genetic background have individual personalities. They behave differently, and it's probably we always know genes versus environmental interactions on behavior, but that clearly shows that there are environmental influences and influence the development of a fruit fly and affects its individual personality. And that's certainly the case for not only humans, but many other animals as well. And that's going to affect learning, I think, for sure.
Dan, did you want to comment on that topic too, or Gwen? Or Dan was first, so Dan.
I had a different topic. Go ahead.
My topic was also a different topic.
Okay, let me before we jump to
add one comment. Then it was just I totally agree, and I think that much like we've seen that variability in neural noise isn't really that much variability in neural noise. It's really related to movement. I think variability in animal behavior is really key. And I think tools. I mean, sorry to be like a tool builder here, but I think like new tools for like new technologies to measure more neurons. Measure behavior at a better scale. Like we're really in a golden period for like from a neuroscience perspective. Of like having this incredible toolkit, right? Because before we were limited, like the reason we went into the lab and were reductionists is because we could look at one cell or a handful of cells, but now we can look at thousands of cells, or like, like what Gwyneth is doing, like you can look at the whole organism and see what it's doing. And now you even have a connectome. So I'm actually really, really excited to see what like the Drosophila crowd does, because that'll really lead the field in many ways, I think, so.
Dan, you wanted to riff on that?
Yeah, actually that connects nicely. I had been studying lizards swimming in sand and other higher creatures for years and years and years. And it was Tom Daniel who said, why don't you study something that people care about? He said, go study the nematode. So I said, well, how complicated could a nematode be? It's only, as people will tell you, has 302 neurons. And so we started looking at it. And one of the nicest surprises we had is just how much more capable the lowly nematode worm is than our best robots, limbless robots on the planet. And I think in terms of, simplicity and doing more, and Mackenzie, your comment about, they can do more sparked this. I think that just the diversity of abilities is really hard to appreciate. These worms are just incredible. They go anywhere. And I just want to make that point.
No, I think that's a great point. And I should qualify my statement being that we as humans can teach them things and then challenge them, where I don't know how to teach a nematode, right? So I think you're absolutely right. Because even people are like, oh, mice, they can't do much. But actually you can teach them to play video games, right? Like it's totally possible. So I agree with you.
There are definitely ways to teach nematodes. Associative learning things. There's a lot of work in that area as well. It's maybe not as cute as teaching a squirrel, but yeah,
and we have video games for the moths.
All right, so this is like a game-changing conversation for me. This
exactly.
Is great.
Very good. Tom would be proud. Gwen, did you have any follow-up?
I'm gonna try to bridge from there to the topic that I wanted to revisit, which is a theme that I think is important to 4DCP that's come up several times, but also in this session, which is the importance of making measurements in vivo, or as Jimmy even extended it, like into the wild. You know, I think that there was a sense at Janelia that it's an exciting time to try to go beyond just, you know, Petri dish measurements and actually explore more complexities of the real environment. And so I guess, you know, first of all, I should say, I'm not advocating that we throw away lab studies. Obviously, I think, you know, there's been some kind of sort of consensus amongst this group that a variety of approaches are useful and you need to kind of go back and forth. But it seems clear that we lack some of the tools we need to really make critical measurements in vivo or in the wild. And I was just interested in kind of throwing it to this group to dig in a little bit more about, you know, maybe highlighting particular measurements that you would want to make, you know, even thinking back to, you know, some of the cellular or subcellular phenomena, you know, if you want to measure them in the actual animal.
I mean, something I've been thinking about in conjunction, and this is probably more relevant to the tiny transparent organism group, is not, I mean, I'd love to be able to watch a nematode crawl from one side of a compost heap to another over days or whatever. But I think in terms of like microstructuring complex environments, there's been a lot of advances in that recently. And I wonder just how far, at least for those class of organisms, and there's others I think you can put into that bin, how much of the wild can you bring into the lab? If you can really characterize local microbiology using tools from soft condensed matter, et cetera, and, you know, innovations in microstructured environments, how much meaningful complexity can you convincingly bring into your microscope or whatever? Something I think worth thinking about, especially with smaller models.
That's interesting, because that almost says that one wants tools to measure the environment more precisely so that you can reproduce it. Yeah, it's interesting.
Bob?
Mackenzie and others, I want body cams for every individual squirrel, and looking at just our GoPros that we leave on, that alone is a study of understanding the variation and capabilities. So I want a global cam too that tracks everything from the sky. At the same time, I want one on every individual, besides monitoring everything else that you could possibly do. And I think that it would just change our view of things, you know, incredibly. It is true that, you know, that there are general responses no matter what happens in the individual variation, that especially when you push to, you know, the biomechanical limits of things, and just like that you see that variation. But there's so many reasons, I think, that from past history, at least in development, that you see differences in, big differences in decision making. And you see these personalities, and these are real effects. We can identify, we put a little dye, we're allowed to, on the squirrels to identify the name them, but you don't need to do it. Yeah. It's incredible, the personalities that they have. And so just like, track everything. I mean, I think that 15 years, I'm sure we can do that, right?
Yeah, no, I think it's closer than you think. Yeah, I'm super excited about this. I also love squirrels and just fascinated by watching them in the backyard. It's kind of sad in Europe, they're like more shy. So I kind of miss the squirrels in Boston, but but you're right. So yeah, I'm really fascinated by this question. And so one thing that we've been doing is like actually just watching animals from birth until adulthood. Like it's like the Truman Show for animals, right? Like we just film everything and we want to know everything about them. And so one of the things, like you said, is you can really start to get fingerprints of these animals. So we're interested really much in this idea of behavioral fingerprinting, but even the computer vision tools are getting very good to do identification. So like you can re-ID an animal with, 99% accuracy just based on visual features that are not like key point tagged in our latest software release. And so I think these types of tools for us are just like, we developed a tool because these cool questions come up. And so I absolutely agree with you. I think it's going to get there within much more than within 15 years. So I think we're
ready. I think
awesome. Yeah.
so many different creatures, right?
Yeah, exactly. Yeah. So no, animal identification is super interesting in general. That's a huge field and it's really fun, but I'll save it for later.
All right, that was really great, guys. I'm sorry to interject. We have to finish up because we're at the end of our discussion session, but we're going to have a whole closed session at the end, so we can revisit these ideas then. Janine, you want to take over?
Great. Thank you guys so much. That was a great session. Let's take a 5-minute break right now, and we will resume around 3:12 p.m. Eastern with session 8. See you then. All right, I think we will go ahead and get started with the last session. And to do that, I'm going to turn it over to the moderators, Gwyneth Carr from Janelia and Dan Goldman from Georgia Tech. So over to you guys.
All right, we have made it to the final session of a very exciting workshop. And I'm really excited. This session, I think, is going to bring things full circle for us. I think we've got some talks across all of our different scales and highlighting some of the themes that we've talked about thus far. We'll get into interaction with the environment, some synthetic approaches, and then some development at the cell scale. So without further ado, I'm happy to introduce our first speaker, Michael Levin from Tufts University.
Right. Thank you so much. I want to thank the organizers and the other speakers for an amazing workshop. This has been really inspirational. So what I would like to bring up are the following points. That there's been this real focus on model systems and a kind of fidelity towards realistic or standard anatomy and behavior. And even non-model system organisms are natural, highly evolved kinds of systems that can obscure some really key knowledge gaps that we now have the ability to address. And this is because of the familiar outcomes of standard development and a kind of really emphasis on selection histories as an explanation for form and function. And I think that we can make some really important progress in 3 ways that I'm going to try to illustrate. First of all, by embracing chimeric and bioengineered novel organisms. So the idea is to look at these things not as sort of poor implementations, let's say, organoids and things like this, not as a poor implementation of the full feature, but actually life as it can be. Yeah, and to explore the full option space for biology environments that never existed before and learn more about what's possible for biology. Also focus on plasticity and the divergence from genomic defaults. And these are implemented by physics and physiology and probably other things. And really to try to mine other fields like cognitive and computer science to understand, for example, morphogenesis as a kind of collective intelligence that's solving problems in a variety of novel spaces. So these are the themes that I'd like to talk about. So I will start with, I'm going to show you a lot of frog and flatworm things. I want to start with a mammal just to be clear that this is not something that's only for so-called lower animals. So this is a phenomenon well known to deer farmers. This is called trophic memory. And what happens is that deer every year grow these amazing racks of antlers. And in certain species, if you come along with a knife and you etch a little score, the damage into the region, that antler rack makes a little callus. They fall off that year. The next year, and roughly for the next 5 to 6 years, they grow back with an ectopic tine at the same location. So think about what that means if you were trying to make a molecular model of this, right? That there are cells at the base of the scalp that have to somehow remember the location of damage and then guide the morphogenesis in this way in subsequent years, right? So really already we can see that with the tools that we usually deploy for these things are probably insufficient. Now, let's talk in general about predicting 3-dimensional anatomy. So here we have a Xenopus laevis, a frog tadpole. They have no limbs at this stage. Here we have axolotl embryos. They do have limbs at this stage. We have the genomes for both species. And so now we can pose the following question. Here's a frogolotl. So what we do is we make these frogolotls, which are part frog cells, part axolotl cells, and we ask a simple question: is this thing going to have legs? The point is that ahead of time we have absolutely no idea, even though we have the genomes, because we really don't understand the algorithms by which these cell collectives make decisions, such as what large-scale structures they're going to make. We simply can't know at this time. And so This is even more sharply seen in examples like this. So these are planaria. These are flatworms that regenerate every part of their body. And so this is a species where these green cells will regenerate a flat head. And when that flat head is complete, they stop. And this is a species with a round head, and these cells regenerate a round head. And when that process is complete, they stop. So what we can do is put some of these cells, transplant them into from the flat-headed form to the round-headed form and make a chimera. And we let the cells move around and get comfortable. And then we ask a simple question. When we amputate the head, what shape are we going to get? Is one shape dominant to the other? Do we get an in-between shape? Or in fact, is regeneration never and remodeling never going to cease because none of the sets of cells are happy about what the stop condition, that the current head that exists, they're just going to continue to remodel. So we really, these are major questions that we really don't understand. And in our lab, we saw that this process has a kind of really interesting intelligence to it in the following sense. Tadpoles have to become frogs, and in order to become frogs, they have to rearrange their craniofacial organs, right? So the eyes have to move, the jaws, the nostrils, everything moves. And so we made these Picasso tadpoles where everything is in the wrong place. The eyes on the back of the head, the jaws are off to the side, everything is in the wrong starting position. And what we found is that these animals still largely make perfectly normal frogs because all of these different organs, they start in the wrong position and they will move in novel paths. So sometimes overshooting and having to come back. But basically what the genetics gives you here is a not hardwired movements, but an error minimization scheme that continues to remodel until a correct frog face is formed. So this sort of, if we had a robotic swarm that was able to do this, we'd certainly call this a collective intelligence. They're able to make up for deformations and changes of circumstance to get to their correct region of morphospace. They're able to solve that problem. And so one thing we really, I think, need to do is to really understand these feedback loops in this process of anatomical homeostasis. We need to understand what happens when the body is shifted away from normal morphology, be it by mutation or injury or teratogens or whatever, and to really understand how the system actually stores the set point, okay, of what the correct morphology is to be able to reduce that error over time. And so, all these important questions: what do the cell collectives measure? How do they store it? How does this plasticity? It has really important implications for evolvability and what happens when you make mutations. The fact that you have actually subunits that can make up for certain changes. And so, in order to study this, we took inspiration from another multicellular system that is really good at flexibly pursuing specific goals, and that, of course, is the brain. And so, in the brain, what happens is that you have these electrical networks that at the level of the hardware made of ion channels and electrical synapses and so on. And they underlie this really interesting and flexible software that manages the information flow. So it turns out that all cells of the body do this. Every cell has ion channels. Most cells have these electrical synapses, exactly the same machinery. And what you're seeing here is a voltage dye time lapse looking at the early cells in a frog embryo as they coordinate with each other as far as who's going to be anterior, posterior, left, right, and so on. And so we developed some tools to manipulate this in vivo. So this is all done by manipulating ion channels, either by mutation or optogenetics, so no electric field applications. And you can do interesting things like you can induce an eye-specific bioelectric pattern in the early embryo, and you can induce it in the gut. And so you can tell these gut cells to make an eye, and they will do that. In fact, you don't have to get all the cells, you can get some, and these cells will recruit their neighbors to make a nice lens or whatever else. These ectopic eyes have all the right tissue layers inside, so you can actually manipulate morphogenesis at a very modular level. You can say build an eye here, and you can do this. And one interesting thing is for the regenerative applications, when you take a frog, which normally doesn't regenerate its legs, and you ask it to regenerate the leg by providing a particular bioelectrical state at the wound, when it does this, you can see that eventually the leg is quite respectable. This is a pretty good looking frog leg right here, and it's touch sensitive and it's motile, but it gets there through a path through morphospace that's completely not the way development works. So this is an early version of that leg, and you can see this is not how frogs normally make their legs at all, and yet it gets to the correct pattern. So this plasticity, this ability to get to the correct region, is interesting. And because it's bioelectric in nature, at least partly, it is rewritable. So in planaria, we have this electrical gradient that tells this fragment where the head and where the tail should go. And if you manipulate that electrical gradient, you can make double head worms, or you can make no head worms. Here you can see also very interesting behavioral model system for two brains in the same body. Once you understand this electric circuit that sets up this gradient, you can make these changes. Now we can model and simulate this, and then we can ask this question: if you have a two-headed animal and you, in plain water, no more manipulations of any kind. Remember, they're not induced genetically. This is all, brief pharmacological ion channel drug exposure. You get rid of the primary head, you get rid of the ectopic secondary head. All you have is a nice normal middle fragment that has the normal wild type genome. Ectopic tissue is gone. You say, what will happen in plain water? Well, surely you would get a normal worm back. That, in fact, is and the modeling actually told us that, yeah, the single-headed animals are a stable attractor in the circuit space, but there's another stable attractor, which is the two-head. And so when you do this experiment, you find out that after you keep cutting them, you will get in perpetuity two-headed animals. There's nothing genetically wrong with these. Their genomic sequence is completely wild type. And so that information about how many heads they're supposed to have and where the heads go is something that is stored at this physiological level. You can write it and rewrite it. So we can take the two-headed animals, bring them back to the one-headed state, and so on. Not only can you decide head number that way, you can decide head shape belonging to other species. So here's a triangular species, you cut that head off. If you perturb the electrical network, you can get heads that are flat, like a P. felina. You can get heads that are round, like an S. mediterranea. The frequency of getting those heads is proportional to the evolutionary distance between these species, about 150 million years between this guy and these. The insides are changed as well, shape of the brain, distribution of stem cells. And so you can explore these attractors in morphospace without changing out the hardware by controlling the electrical computations that this network makes. And in fact, you can change the body plan entirely. You can make these cylindrical animals and so on. So these body plans, and just a couple more slides here, these body plans are not only anatomically different, but what's amazing is they're highly functional. So if you make tadpoles with eyes on their tails and no eyes in the head, these animals can see perfectly well. We have a machine, we test them on visual learning tasks. They can see out of this. So moving the eye to the tail, the eye connects to the spinal cord. It does not connect directly to the brain. No problem. The animal can adapt to this perfectly well. And you can sort of take it to its logical conclusion and make organisms that have never existed before. So this is, we call them xenobots. They come from the skin of Xenopus laevis. You can see them moving along. They can walk. If they have legs, they will walk. Who we model this. If you alter their bioelectrics, you can convince the cells not to come together to form this, and they sort of spread out and disband. They have all kinds of interesting behaviors. They can go in circles. They can patrol back and forth. They have spontaneous behaviors. So you can see here that this is still water in a maze. There's no flow. They turn the corner without bumping into anything. Here it decides to turn around and come back. So some kind of internal dynamics. They regenerate. So if you cut them, if you damage them, they will heal back to the new xenobot shape. And they have these really interesting calcium dynamics. Remember, there's no neurons here. This is all epidermis. This is all skin. And so they have group behaviors, meaning collective kinds of behaviors. They have individual behaviors. And we're currently testing them for sensing, for preference, for learning capacity, and so on. And in fact, you can make little tiny ones that have much fewer cells, such as this here. They also have interesting behaviors that you can study. So really minimal model systems for novel plasticity during morphogenesis and behavior. And we can now do this, of course, bioengineers can do this at all levels. So at the cellular level, at the genetics level, at the cellular level, you can, and all the way up, you can make chimeras and novel creatures with their new worlds that they inhabit, which might be instrumentized, they might be, whatever. So just to finish up, my claim is that by embracing the fact that we can make novel organisms that are not meant to be models of something else, let's say the human brain or anything like this, we can explore something very interesting, which is how evolution solves problems using the same tricks, in particular, a very brain-like electrical network, to solve problems in transcriptional space, physiological space, morphospace, and behavioral space. So I would just like to thank, these are the people that did the work and our funders, and thank you.
Great, thanks so much, Mike. A really great and thought-provoking talk there. So while we have a couple minutes for just a few questions, while those come in, I just wanted to ask you a sort of higher level question about your general thesis. So you're sort of proposing that you know, a scientist could use these kinds of chimeras or these not sort of natural combinations of things to probe, you know, some kinds of physical or other kinds of biological phenomena. But I guess earlier in this workshop, we've heard people argue to use sort of natural variation for exactly that sort of same idea. So I just wanted to hear your thoughts on specifically, you know, and I guess the idea would be that natural variation itself tries to fill every niche. So you're already trying to cover a lot of these different sort of combinations. How, you know, what more do we get from chimeras?
Yeah, so look, I certainly don't think it's an either or situation. And I think studying existing model systems in their ecological environment is hugely important, right? But still, every standard model animal is part of a lineage of a long history of selection forces that shaped its behavior and its function. And that means that what we can do now that's slightly different is put together emergent bodies and primitive minds that don't have that history. So when you think about that and you ask, okay, why you've made a novel synthetic creature, it has certain morphogenetic features, it has certain behaviors. Where did those come from? If it doesn't have a long history of selecting for specific behaviors and morphologies, what that means is that there's some really interesting physics and physiology that can provide those basically on the fly, right? And we need to understand the emergence of those. And those are often hidden by these long histories where we're kind of, well, of course it's a zebra, you know, zebrafish eggs make zebrafish, and they always do. And so there you go. And it's because the fish needs to swim like this and like that and so on. And so the idea is that we now have the ability to watch these things emerge in a very rapid time frame, where the answer to where are these coming from is just different than it's going to be for a standard model species.
Okay, great. One last quick question. Where and in what form exactly is the homeostatic set point stored?
Yeah, so we only have it really nailed down to any degree in one model system, and that's the planarian. So the thing about the planarian is we have actually now observed you can, using these voltage dyes and some other techniques, you can literally see the bioelectric pattern that's going to distinguish a one-headed worm from a two-headed worm from, there's actually a couple of other types. So that standing electrical pattern is literally a kind of memory. I mean, because it uses the same mechanisms as memory. It is a kind of memory that stores this information, and it's stable. Once you switch that circuit, it stays until you go and you switch it back, and you can see it and you can manipulate it. So we think that is one example. There may be others. There may be physical forces and patterns of stress and strain and who knows what else, but there's certainly an electrical kind of memory that keeps it.
Great. Okay, well, in the interest of time, we're going to move on. Mike, you do have another question in the Q&A, if you could take a look at that.
Will do.
I'd like to introduce our next speaker, Chen Li from Johns Hopkins University.
Oh, sorry. So thanks to the organizers for the opportunity, and to everyone for hearing a little bit of our work and my thoughts on this topic. So animal locomotion. Yeah, so animal locomotion emerges from the mechanical interaction controlled by the nervous system with real environments, very often involving transitions between different locomotor modes. For the study of flight and swimming, we have a long history of aero and hydrodynamics entire field with many well-established tools and models to help us understand how locomotion emerges from the interaction with the complex fluid environment. For terrestrial locomotion, we also have several decades of very nice systematic studies, but by necessity, these have started on relatively simple, flat, rigid ground. And when it comes to more complex terrain that we see in nature, such as granular media like sand that can flow, or 3D terrain with lots of obstacles, then we have a relative lack of tools to study these interactions and understand or even predict how movement emerges from interaction. So in other words, we need to begin to create a new field of thermodynamics to try to understand terrestrial locomotion in the real world. And this has really been the focus of my research, starting when I was still a PhD student in Dan Goldman's lab. Really, this was pioneered by Dan's lab using granular media as a model system. And over there, I focused specifically on legged locomotion on sand, creating tools and performing systematic experiments. We eventually created the first model capable of predicting legged locomotion on granular media. And after my PhD, I began thinking about how to expand thermodynamics into complex 3D terrains such as these with many heterogeneous large obstacles. So when we look at how our knowledge of mechanistic and even predictive models of terrestrial locomotion, we can see that we have several decades of study which have helped us reveal how to generate and how to stabilize near steady state locomotion, such as walking and running on flat ground and when you have perturbations from obstacles. But when it comes to 3D terrain like these, shown by a couple of videos here, you can see that it's not clear whether this kind of steady state limit cycle like dynamical systems theory or concept can still apply here. So we have a relative lack of understanding locomotor transitions. So I first began thinking about this when I was doing my postdoc with Bob Fo. And in Bob's lab, we have all these cockroaches that are native to tropical rainforests that have to go through these kinds of environment all the time. And you can see this seems like a formidable problem. You have a suite of heterogeneous obstacles. So how do we begin to study this? So at the time, we decided to focus on one type of obstacles, which are dense vegetation. And by using grass as a model terrain, we created artificial grass-like beams that can enable controlled and repeatable experiments. And we found that the animals make robust transitions across many different modes, forming these multi-pathway locomotor transitions. So over the last few years, a major part of my lab's research has been to try to understand how animals can deal with a diversity of challenges which we abstract from such natural complex 3D terrain. And we're hoping if we can understand how their locomotor transitions emerge from interaction with these distinct types of obstacles, we can perhaps put everything together to understand the formation of multi-pathway locomotor transitions. Okay, so towards that goal, we have been developing laboratory tools to control and vary the terrain parameters to measure movement in 3D. And also using these, we perform systematic animal experiments. We also use robots as physical models to not only control, but vary locomotor parameters to test hypotheses and try to discover if there are general principles. And finally, through all these, we can then begin to create physics models that describe how animals or robots interact with the environment and how they can control such interaction to generate locomotive transitions. So through all of these, it turned out to be quite surprising. We didn't expect this, but when we recently looked back at the suite of studies we have done on this diversity of model systems, we found that there are surprisingly general neuromechanical principles. We found for all of these systems, there turned out to be highly stochastic, but also really stereotyped locomotive modes and transitions. And this is because there's an underlying potential energy landscape which had distinct basins, and the system's state is strongly attracted to these basins because they keep destabilizing themselves through the cyclic interaction with the environment. And the locomotor modes, the locomotor transitions then manifest as the system makes transition across potential energy barriers from one basin to another. And this conceptual framework had allowed us to understand a variety of strategies that we have discovered empirically through experiments as one of three categories as shown here. So our next step is we hope to try to begin to piece these together to understand and even predict how multi-pathway locomotor transitions emerge from neuromechanical interaction with the environment, analogous to what people have done for a few decades, understanding how multi-pathway protein transitions emerge from a free energy landscape describing the interaction. Yeah, so towards the end, I just want to talk a little bit about my thoughts on this topic. So we know that animal behavior emerges from highly complex coupled interactions across many different systems, from very small to very large scales through the organism itself as well as its environment. And on the larger side, we have the field of movement ecology, which tries to quantify and describe animals' movement in nature. So we have very nice bio-tracking techniques that have been developed to help us determine what the animals are doing in the wild. But a challenge is that in these studies, the animals are effectively point particles, and it's challenging to study the detailed neuromechanical interaction. On the other end of the spectrum, we also have a huge field of neuroethology to understand the neural basis of behavior, with a lot of nice advancement in neuroimaging and also using optogenetics and other tools to even control the organisms. And also there's recent very nice advancement, as some of the talks have already shown, to apply computer vision and artificial intelligence and learning techniques to quantify in great detail, high spatial resolution, and over very long temporal scale, the locomotor behavior transitions of organisms. But very often, in order to do this through computer vision, you have to study the animal in relatively simplified environment. Yeah, so just some of my thoughts. So first, obviously, we need interdisciplinary and integrative training and collaboration. And also, I think that we really need to study animals more in realistic environment, to which we want to develop more laboratory tools, platforms to mimic nature, as well as to perform lab level measurements in the field. And also for these, we need better sensors to sense not only the organism, but also the environment, and not just the internal signals and the kinematics, but also the dynamics. We also, as many of the talks have already shown, it'll be very useful to study more diverse and ideally highly performing organisms for diverse environment, and finally to kind of move beyond tethered and two-dimensional treatments to study free moving animals in 3D. And as our own work and among many others have shown, that robotic physical models or other kinds of active synthetic systems can be a very useful tool to complement biological studies, because they have several advantages. You can generate relevant locomotor behavior through much simpler design, actuation, and sensing, which are easier to analyze and model. And also they can be easier than biological organisms to control parameters and vary parameters, to test hypotheses and to discover principles. Further, they can be studied in open loop first to isolate passive mechanical effects from active sensor feedback. And finally, unlike theoretical models, which have to be very carefully validated and interrogated, they actually enact and do not model the laws of physics. So they offer advantages there. Yeah, so just a couple of examples in other locomotion studies. So here's some nice work done by Jimmy Liao's group, looking at how to understand the dynamics and mechanical sensing of swimming in turbulent water by combining animal studies, robotic physical models, as well as sensors inspired by the sensors in animals, as well as work done by Simon Swanberg and others, as well as Stacy Coombs' group, to look at realistic environment, how animals can track flower and navigate obstacles, and even like performing these experiments in field stations. So with that, I'd just like to thank my group members, a lot of colleagues for discussion and our funding. And thank you for your attention, and I'll take questions.
Great talk, Chen. Thank you. All right. We're running a little behind. So in order to catch up, I'm wondering if there's one quick question from the audience Otherwise, we will maybe save discussion. I see comments coming in a little bit late as people think about things. So we may save all of our thoughts for the discussion coming up soon. I actually have some questions for you, Chen. All right, I think in the interest of time, I might pass things to Dan to get us going with the second half of the session.
Okay, all right. Thank you all. We will proceed with our next speaker, Julia Paci. Can you pull up your...
Let me share my screen. Okay, it should be up.
Right.
Great. So hi, everyone, and thanks to the organizers for an opportunity to speak at this really, really amazing meeting. So I'm Julia. I'm a postdoc at the UCL, and today I want to talk about my project, where I'm really interested in how mechanical forces can be buffered during animal development, and in particular during pattern formation. So the context that I really like to think about is really this idea that when we have development occurring in basically any organism, this is always while we also have the continuous action of varying forces. And just to give you an example, we can think about external forces. And I mean, many examples have already been given in previous talks, but if we think about plants, we know they have to continuously adapt to changing environmental conditions, and these conditions will also ultimately reflect in very different outcomes. And in animal system, again, one can think of many examples, but here I have an example of zebrafish heart development, where we know that the developing tissue is continuously interacting with other factors, such as, for example, blood flow, blood circulation, that in turn applies a shear stress on the tissue. And this is all kind of while development is occurring, and this is changing over time. So to me, this really brings about a picture that we have the presence of mechanical noise. Again, this is all external forces I'm discussing, but these can also come from actually internal sources as well. And in the presence of this mechanical noise, we still observe that developmental patterning and development in general can be extremely robust. So that kind of brings me to the core question I've really been interested in, which is how can we have this mechanochemical signaling pathway that can, on one hand, respond to instructive signals, but they also have to be able to buffer this sort of mechanical noise. And the system which I'm currently using to address this question is the wing disc of Drosophila. So this tissue is later on going to develop into the fly wing, and it grows dramatically while it also undergoes patterning following very well characterized pathways during the larval stage of development. And at the same life stage, we also know that behaviorally, the larvae are basically continuously crawling in order to feed nonstop and power this amazing growth. And the wing disc is not just floating inside, it's kind of like bag of fluid, as you may think, looking at the larvae, but actually it has lots of attachment points, both to the epidermis and to other internal tissues. And therefore, we can imagine that it's going to experience continuously fluctuating forces which are transmitted throughout these days of development. So in order to address this global question, I'm taking an approach that really combines both in vivo and ex vivo manipulations and ideas from both biology and physics. So I think we will have seen many of these discussions of advantages of the different approaches, which I think you will appreciate here as well. And two of the key things I've been thinking about is, on one hand, what are these external forces that are experienced by developing organs in vivo? I feel like we still don't really have a very good idea on what sorts of magnitudes and timescales that this happens. And the second question is more specifically, if we take patterning pathways, how are these actually established and maintained in the presence of these external forces? And to get to that first question, the approach I've been undertaking is one of using live imaging. So what we can do is we can label the tissue we're interested in quite specifically. So what you see here highlighted are the two pouch regions of wing discs. And this is in a freely crawling larva. So then you can image the larva as it's happily crawling on the microscope. And as you can already appreciate from this short movie, even from few seconds of crawling motion, you can clearly observe that there are a lot of deformations and pulling and tugging that is acting on the wing discs as the larva is crawling. So the first thing that we're really focusing on at the moment is from such movies, trying to extract images of strain conformations of wing discs and then comparing this with resting state images. The idea is really to calculate both the magnitudes of these in vivo strains, but also the time scales over which these occur. And then I'm kind of now switching gears a bit and moving to our ex vivo approaches. So as I said, I'm also really interested in this interplay of signaling pathways and mechanics. And of course, this is a very, very complex question. And I feel like sometimes ex vivo approaches can be very valuable because allow us to control certain aspects, certain parameters much more precisely. So what you see here is a tissue stretcher. It's a device that was previously developed in the lab, and this allows us to culture discs ex vivo between two PDMS layers. And then through these, we can basically apply any arbitrary stretch or compression to the tissue. So we can control the magnitude of the stress, but also its temporal pattern. For example, a single stretch or maybe a cyclic one. And by using such a device and such an approach, again, we can have a very good control on the mechanical input to the system. So given this input, then what I'm asking is, well, if we look at live reporters of signaling activity, looking at different pathways that are important in patterning, what kind of response do we see to these different regimes of stress? And these are just like some hypothetical curves to show you that, we might have, for example, a minimum threshold of force that is required for activation, and then different types of dependence on the pathway activation on force. And then quite nicely, what we can also do quite easily in Drosophila is look even more downstream. So we can look at markers, for example, of cell fate, and look at how basically downstream patterns that are guided by these signaling pathways are affected. So basically this allows us to also ask the question whether the ultimate patterning is perturbed by mechanical forces, or actually the signaling pathways themselves take care of kind of having a feedback loop and buffering mechanical noise. And because there's this idea of having a quantitative approach, you can imagine that ultimately what one might be able to do is even reconstruct a whole phase space almost of different responses of a given pathway to forces, where given a certain magnitude and given temporal information, we can imagine ranges where the pathway is going to be completely insensitive, ranges in which maybe the tissue will undergo rupture, and then intermediate ranges where we'll have response and buffering and adaptation to that input. So just to give you an example of one of the pathways and patterns I've been looking at, what you see here are these two parallel rows of cells, which are the sensory organ precursor. So these cells are specified towards the end of larval development, and in the end, they're going to form sensory bristles that basically line the entire wing of the fly. And they're specified through notch delta signaling that occurs through neighboring cells and may also occur at a distance through protrusion. And this pattern, we can actually image it as it's specified over time. So this is a time-lapse movie of a cultured wing disc ex vivo. And you can see that over the time course of roughly 12 hours, we can see all these cells acquiring gradually their proneural state and becoming specified in this pattern. So what I'm currently doing is really taking these conditions and comparing how the pattern is established in control conditions. And then, since this timing is also compatible with our stretcher ex vivo culture system, comparing how development occurs in a case where we have applied an external force, for example, here, a sustained single stretch over time. And then as the last thing, I also want to go back more to the in vivo system. And I think one thing that would be really, really interesting is also to try and actually modify the environment in which the larva is growing in an in vivo way. And there's many ways one can think of doing this. The approach which I'm currently undertaking is basically growing the larvae into PDMS channels. And in these PDMS channels, we can also add all sorts of constrictions and just allow the larvae to freely crawl backwards, forwards, and enter into these constricted parts of the channel. And what we see is that actually when they do that, you see again like an overlay of the channel and the wing discs labeled in red. If you follow the wing discs over time, you can see that over a time scale of seconds, we have these huge deformations and squeezing and pulling of the organs. Again, in an intact larva that is crawling through that constriction. So this is maybe a complementary method that we can use to actually apply in vivo strains on the larvae. And I'm currently following up on what are the short-term and also long-term effects of such in vivo strains onto the patterning and mechanical response of those tissues. So to wrap up, I just wanted to mention one thing which I see currently is still quite challenging in my project, and it's already come up. Clearly, this idea of having, fast but also volumetric imaging over a big, spatial scale in a living motile animal, and we need it to be fast because, again, you can think these are fluctuations in wing disc which are due to the larva heartbeat. And these occur on a very, very fast time scale. And most of the images I've shown you so far have been in wide field microscopy because this is one of the few that allows us to image that fast. But of course, wing discs are actually 3D objects. So it would be much more informative to get the whole 3D information. And you could even ask how they actually interact and attach to all the other organs and the guts and the epidermis, and how all those interact during development. And of course, this maybe connects a bit also with other talks from today. But if we had, such a microscope that allows us to do that quite easily, you could imagine then endless possibilities. For example, there's very beautiful locomotion mutants already characterized for Drosophila larvae that have different mutations in their nervous system, and that causes them to have, for example, a very directed pattern of motion, or basically turning all the time. And you could imagine then having these simple systems and being able to really image and quantify the forces that are experienced in vivo. So with that, I just want to thank both of my labs and thank you for your attention.
Okay. Thanks, Julia, for a beautiful talk. I see questions in the chat. Any questions from live? I see Manu has his hand up. Go ahead, Manu.
Yeah, beautiful talk, Julia. And I think one thing that I wanted to have you comment on, if you push the limits of the stretching to bring it in the yielding regime, we've seen epithelial fractures recently in a physiological setting that are repaired live. And I've always been wanting to see that in more number of organisms. So I'm curious, what is the end state when you stretch further?
Yeah, no, that's a very good question. So I have to say, so far, I've definitely used the stretcher to challenge the tissue to ranges of strain that are certainly beyond physiological range, because I do think that it is informative, as has been said previously, also to kind of bring the state maybe to its more like limit in order also to learn of how it works. So far, I haven't really brought it like to the level of having ruptures. I think that's like a very exciting idea, and we do know that actually wing disc can repair pretty well. So some other people in the lab really work on wound healing. So actually, I think that would be something really cool to try. I haven't tried it yet.
Okay, I see a question in the chat, not the chat, whatever it's called, the Q&A. Do the strains, and if so, how, change the Lyme PCP in the wing disc?
Yeah, so this is something we don't know yet, and definitely it's one of the things that we also want to look at. So right now, I'm really looking more globally at really getting an idea of what kind of strains and also what kind of timescales, because there's many timescales overlapping at the same time. There's going to be, this kind of peristaltic crawling, and then much faster timescales, and then turning motion. So all of these are kind of superimposed with each other. And so far, I haven't really in more detail looked at the specific effects on, for example, specific, boundary segmentation or patterning in the wing disc. But I think that's definitely a very interesting point.
Okay, I think that...
Hey, Dan, actually, there's a question from Claudia.
Why did I miss
Claudia.
that? Sorry.
Oh, hi. Really beautiful talk, Julia. It's really cool system. I I was wondering, and this is kind of inspired by watching your movies of the larva crawling and then putting them in constriction. In your device, is it possible to push and pull so that like one side of the wing disc can be kind of constrained and the other one can be stretched, like the larva were like turning a corner? Is that something that's feasible?
Yeah, so I mean, for sure, there's lots of people that have worked with this kind of PDMS devices, and there's definitely approaches where, for example, you kind of like push it in or you pull it in. So far, I've always preferred to basically have them go into the like crawl into the constriction by their own will. And actually they can squeeze into really, really small channels. So what maybe the thing that would be closest to what you suggest is I now have different devices where I kind of like a snake-like channel. So they're kind of crawling into like straight and then turning 90-degree turn motion. And so in that case, you might see the type of deformation that you're talking about.
Oh yeah, I guess I'm thinking for your ex vivo one, where you take out it and you stretch the wing disc. Can you like right now I understand you're stretching them like this.
Yeah.
But can you like stretch and compress on one side? Because that would be almost like, it seems like you can do better imaging in your device, these devices, than you can in the live in the animals. And so that would be analogous to turning a corner.
I see what you mean. I mean, with the current setup, I don't think we would be able to do that because it's kind of like uniaxial. So what you will be able to do is apply it on one direction, or you can mount it on the orthogonal direction and do the same thing on the opposite. But you wouldn't be able to necessarily push and pull at the same time. I think you would have to think of a different like smart design to be able to do that.
Cool, thanks.
I see Adrian has his hand up. Go ahead.
Yeah, hi Julia, yeah, very fascinating talk. So I have a question. So when you are ex vivo, when you apply strain, do you only apply static strain or do you also apply time varying strain? Because actually, like, this is something we have noted. So in the talk I've given, I've only shown the work we did on static strain, but basically applying time varying strain has also impacts on both the transcriptional program, but also on the remodeling of the tissue at the cellular and multicellular level. And I was curious to see if you were observing that as well.
Yeah, no, absolutely. I think that's a super good point. And for simplicity, most of the things that I've started from are like single stretching, just because it's kind of like the simplest first test to do. But what we know actually is that if you do that and you keep it over a very long time, actually the tissue is going to remodel. So it's going to dynamically remodel. And towards the end, it's not really feeling much strain anymore because it's just moved, adapted to a new equilibrium shape. So actually it's absolutely important to do, as you say. So if you actually want to keep a very high strain over time, you do need to do a cyclic stretch. That basically the tissue doesn't have the time to remodel, and it's always feeling this very high. And of course, another thing which I think is exciting is you could try to mimic physiologically interesting ranges. For example, the period of the striding, of crawling, and I think that would be also quite cool.
Yeah, thanks a lot.
Thank you.
Okay, if there are no further questions for Julia, I suggest we clap for all the speakers again. And we now enter the discussion phase of this session. Again, the session is titled Toward Prediction and Control of Living Systems Across Scales. And Gwyneth and I will, I guess, lead a discussion among the participants and speakers. We'll wait till all are up on the screen. Chen, or you want to? Ah, there it is. Okay, and we will conclude at 4:10, after which point we'll have a 15-minute break, Janine, and then regroup.
Sure, that sounds good.
Okay. All right. So I want to ask the speakers, any common themes you see in your talks in this session? And given that we're about prediction and control of living systems, this is sort of wrapping up the end of the workshop. Anything that jumps out?
I mean, for me, one thing that comes up, I mean, throughout all these sessions is really this idea of what do we want to do with like reductionist approaches, and, you know, this like in vivo, ex vivo, realistic, less realistic. And this is something that I also wanted to also like ask Chen, you know, when you want to simulate this realistic environment, like how realistic is realistic enough, and what are some criteria, what do we think are good criteria of where we set the bar for that? I think this is like a really, really hard question, but it's a really crucial one.
That's a great question. Yeah, I don't know if there's any like formal criteria there. What we very often do, like as we try to create a model terrain or a model like kind of robot, is to build them so that they display kind of a similar behavior. For example, in one study, we looked at how these cockroaches, when they go through complex terrains, very often for them to get flipped over, and they need to right themselves. When you look across many terrestrial animals, this turned out to be highly strenuous. They really are struggling and try to do everything they can to flip back on their feet. But when we look at how robots have been developed to do this, it is very often using all these kind of impulsive actuation to just kind of throw you high off the ground and then fall onto the ground to self-right. So we actually, in an earlier study, we had made a robot which can operate that way. But later when we want to use this, and that was just more of a bio-inspired kind of robot design study, but later we want to better understand this physics principles or neuromechanical principles of such really highly strenuous self-righting. So we actually had to purposely build our robot so that it is very difficult to self-right, and then we can use it as a model. And that's actually something we got asked by one of the reviewers who we think is probably a roboticist. He's familiar with our previous work. So he was asking, oh, what's the point? Why are you building this robot now that struggles? Like, I don't really see why this is useful for robots. And that's kind of exactly why we do that. So I think you definitely have to make some kind of simplification. Otherwise, it's just too complex to study, but you want to maintain the kind of essential behavior that you try to study. Yeah, so I think that kind of this idea can probably also extend like that. Like when we build model terrain, we also try to, it has to be simplified, but also, for example, in all these obstacles, we make them large enough so that the animals can no longer simply walk around them, like avoiding obstacles, which is what we are trying to understand.
Margaret, Mike, do you want to respond, or did you want, Margaret has the question. I can.
Yeah, please go ahead.
Go ahead, Margaret.
Yeah, so my question is for Mike and Julia mostly. I mean, so this idea that this bioelectric circuit is a way of giving plasticity to tissue is a really interesting idea. Like, how do you think, and presumably there's going to be other properties that could behave similarly, that allow for long range information that can be reversible and encode information at longer timescales. In terms of trying to figure out the rules of learning, like, you know, think about it as like a spin up and spin down system of like how things turn and store information. Do you think can you do this in like model epithelial tissues? Do you do this at the single cell for single cell mechanobiology? Like, what approaches do you think you can do to isolate which of these parameters are going to help you encode memory and information, allow for information processing at the tissue scale?
Yeah, I mean, for us, and I've seen this, so I'm certainly not claiming that bioelectrics is the only way this happens. So I've seen people do this in the cytoskeleton and all kinds of other structures. For us, the interesting control properties that we see are multicellular. So the bioelectric code that we're working on is not a single cell property. In fact, the voltage of single cells almost never matters. It's the pattern across large groups of cells, and specifically boundaries, you know, places where there's cells of different voltages where they're next to each other. That's where a lot of this happens. And yeah, you can certainly do this in vitro, and you know, we've set it up in cell culture, and Adam Cohen at Harvard has some beautiful work on this stuff. yeah, all the tools are there. It's really, but in vivo is where whether in artificial or natural organisms, in vivo is where you really see the shine, because the outputs are so much more complex than what you can see in cell culture.
I mean, so in the vivo, in sort of the synthetic organisms you're creating, do you have ways of identifying like how the rewiring takes place?
Yeah, so the merger, so I kind of talked about three different things, right? So the merger of bioelectrics and the synthetic organisms hasn't happened yet, in the sense that we don't even have a paper on this yet. So yeah, this is like just very early days. But yeah, in vivo, what we do, and the frog is super convenient, the worm is, you know, somewhat tractable. You can use optogenetics, and you can track the voltage, and you can lay down light masks of different patterns, and you can look at what's downstream under different circumstances. You can give them experiences, morphogenetic experiences, and you can watch the memories form and remodel and so on. All of this is visible.
Yeah, it's super cool. Thank you.
Let's see, Adrian is next, and then Jimmy had a question, and then we'll get to Ed.
Yeah, thank you. Another question for Mike. Actually, I'm curious about the links between mechanics, and more specifically tissue mechanics and bioelectricity, because like you can construct mechanochemical models of tissues where cell volume regulation plays a crucial role in determining the mechanical properties and behavior of the multicellular tissue. So by controlling the voltage or the bioelectric pathways with pharmacological perturbation, you could in principle also control the mechanics of the tissue. So I'm curious to hear more about that, and you could use bioelectricity to perturb mechanics or probe mechanics, and the opposite, or you could use mechanics to probe bioelectricity in tissues or small organisms. Thanks.
Yep. Yeah, they definitely work together. Celeste Nelson at Princeton is probably the best person on this. She's got some really nice work looking simultaneously at the relationship between things in cells at very high resolution. So we don't do too much in the biomechanics aspect, but she's got beautiful work. So
I think Jimmy was next, and I've asked him to ask his question instead of me reading it.
Sure. This is to get to Julia and Chen's kind of discussion. I was thinking, could there be an analogous spike-triggered average approach? This is like a neurophysiology approach where instead of applying a stimulus like a sine wave that's easy to analyze or what you think is important for the animal, you give it a white noise stimulus. So basically, you know, you throw everything but the kitchen sink at it, and then you look to see where the neuron, for instance, is spiking. So can you give the animal the real environment and then somehow look at its behavior to extract what's important to it?
Yeah, I mean, I think that, again, like right now, I wouldn't really know like exactly which tools to use to do that, but I think that might be again, I come from like the physics world. I'm a physicist by training, so I like to think of, you know, just like applying things to the tissues in a very controlled manner. But I do see your point, and I think that, yeah, it's true. And as we've been saying, since our approach is actually extracting this information-rich data are getting better and better, maybe we're getting to a range in which it is realistic to give, as you say, like a white noise background and still be able to interpret something like valuable on the system out of it without the need of like precise perturbation. I think that's a very cool idea.
Jen, do you want to respond to that real quick before Ed's question?
Yes. So in our studies, we have, so as a first step and kind of following a lot of successful examples to first try to understand the locomotion in the more feed-forward limit. So that's why we, in all of our studies so far, we have relied on studying the escape response, where the animal is bandwidth limited, that their movement has kind of is more affected by passive mechanical interaction. But we did do some preliminary observation. When you put the animal into an arena with these large obstacles, they would still display similar locomotor modes and transitions, but the probability distribution appeared to be, we didn't measure that systematic, but it appeared to be be changed, to be modified. So in that regime, their behaviors seems to be much more dominated by their sensing and then decision making based on the sensory information. Yeah, so I think it really we started from this just to kind of have a basis of understanding the more passive interaction. That's also why we run our robots in the feed-forward limit. But certainly once you have this basis, then it is important to begin to understand that other end of the spectrum.
Okay, I think that we're running short on time, but so we'll close with Ed's question and then we'll take it to break.
Okay, I'll try to make it short. This is for Mike. So I've always been really fascinated by the idea that developing systems seem to go through a series of states that are sort of dynamically stable states, and that when perturbed, they can go through lots of different trajectories to get to those states. So I'm wondering, in the sort of the artificial epithelioids that you're looking at, do you think that there's some, well-defined set of attractors that define their behaviors as opposed to just a continuous spectrum of possible dynamics?
Yeah, that's a great question. We certainly have not probed it in detail to be certain of the answer to this, but from what I see, it is discrete, because we see certain specific changes that we can make that are fairly clumsy as things go, and they will continue on to a pretty well-defined. And we see this all the time in development and regeneration. We make perturbations that are super clumsy, and the thing cleans it all up and becomes like exactly these very specific things. So yeah, I think it's exactly the same here.
Okay, terrific. I think that we're a minute or two over, so why don't I pass it off to Janine?
Yes. All right. Thank you. Thank you guys so much for a great session. I am going to actually turn it back over to maybe Gwyneth or Wyatt for some closing remarks, but I will say that we will begin the closed panelist discussion in, let's say, 10 minutes at around when it's supposed to start, around 4:25, and I'll resend the link for that. But thank you to everyone for joining us. Thanks to the audience, and I'll turn it back over to any of the organizers who want to make some final remarks.
So since this ends the sort of public part of this workshop, I just wanted to, again, thank all of the speakers and panel discussion leaders for these great contributions. I think we've really fleshed out some interesting themes that we'll dig into a little bit in our next session. But in closing, I really wanted to also thank the audience who've joined us, and to implore you in the audience. We're happy to continue this dialogue with you. Please feel free to reach out to myself, Wyatt, Margaret, Dan, Janine. If you have any thoughts that we weren't able to discuss, we're really excited about this maybe nascent field of sort of mechanobiology, biomechanics, however we want to label it, across scales. So yeah, thank you again for joining us, and please send us any thoughts you have.
I also want to mention, in case anyone is still out there, that all of the talks and discussions are recorded, and they will be available on Janelia's YouTube channel, and they will be linked out from the workshop web page. So you can keep an eye out for those. Thanks, everyone.
Thank you all.
Thank you, everyone, very much.